Diagnostic and, and Therapeutic Solutions. I would like to give you a warm welcome this morning from the beautiful island of Barbados. Thank you for joining us, and I hope that you will find this symposium today very educational. How time flies. I can remember our first annual symposium held in Trinidad 19 years ago, and I'm truly honored to be here this morning to open this, our first online symposium from Barbados, in association with our colleagues in Jamaica after hosting face-to-face symposiums here in Barbados in 2010 and 2016. At this time, I would now like to ask Dr. Damien Cohol, Dean of the University of the West Indies, Faculty of Medical Sciences, Cave Hill, Barbados, to bring greetings and opening remarks. Good morning, Dr. Cohol.
Great morning, colleagues. First and foremost, I would like to thank the CACM, Caribbean Association of Clinical Microbiologists, for the invitation to provide some opening remarks this morning. I was scanning through the participants list to see if I had any of my fellow deans in the meeting as well. I'm not seeing any of them present as yet, but I assume at some point they would participate in today's proceedings. So I bring greetings on behalf of the University of the West Indies, and I also bring greetings on behalf of the faculty. Of course, I lighten the executive membership of the Caribbean Association of Clinical Microbiologists, other fellow faculty members of the University of the West Indies, other faculty members who may not be from the University of West Indies, because I know this is a regional association, and persons who are outside of the states where the University of the West Indies are institutionalized. A warm welcome to you. Of course, a warm welcome to our other specially invited guests, media, and other personalities who are here with us. So let me start off by just highlighting that, one, I'm not a microbiologist. I'm a basic medical scientist. My discipline is pharmacology. And of course, we know the close connection between pharmacology and microbiology because part of the intervention mechanisms that we employ within the healthcare system to deal with pathogens, especially microbial pathogens, are related to treatment using pharmacotherapeutics. So I'm not going to dwell too much into pharmacology. I'm going to try and sit on the boundary between pharmacology and microbiology this morning in terms of my opening remarks. But before I get into that, first, I would also like to acknowledge that the University of the West Indies and this Faculty of Medical Sciences, we celebrate our 75th anniversary this year. Not such a long time ago, the university started as a satellite college of the University of London. In Jamaica, Mona was the first campus before it expanded throughout the Caribbean in 1948. Right? And we celebrate 75 years this year. At the Cave Hill campus, where I'm based, we celebrate our Golden Jubilee, our 60th year. And the full Faculty of Medical Sciences, since being established in 2008, celebrates its 15th year today. Well, not today, but this year. So we have plenty to celebrate, and I'm happy to share this occasion with you. So this is your 19th symposium, and of course, within the spirit of academy and academic pursuits, the Faculty of Medical Sciences wishes to celebrate this occasion with you. The theme of your symposium is Emerging Microbial Threats, Diagnostic and Therapeutic Solutions, which, hint, hint, is up my alley as a pharmacologist. But while the theme highlights emerging threats, we know very well over the past decade that we have had some very interesting microbial threats, which has plagued not only our region, but globally. We even had a pandemic we can highlight dengue, which has been around for some time, Zika, chikungunya, and of course, the big C, COVID-19. All viruses, not to say that viruses are the only ones that count, but all very past, current, and emerging threats around us, right? And of course, within that space, to make it a little bit more interesting and make it a little bit more complex, we have the big issue of antimicrobial resistance, right? Which spans right across the spectrum of microbes, talking about viruses, bacteria, among other pathogenic species. So we can't forget those in moving forward. And as we start to think about emerging threats, we have to think about the potential reemergence of any of these viruses, which has plagued us over the last decade. As we know, the incidence of dengue has grown dramatically around the world in recent decades, with cases reported to the WHO raising from just over 500,000 in, say, the year 2000. And these cases continue to rise to about 5.2 million in 2019, right? So just over a 2-decade period, we're moving from 500,000 to just about 5 million cases of dengue. In the Caribbean, we have seasonal climate, dry versus wet. With climate change, a lot has to be said about that seasonal climate shifting, right? And what we normally see in terms of increment in incidence of dengue, which of course we know is basically transmitted through a vector, the mosquito, right? Of course, proliferating during the rainy season, we might now start seeing that in different aspects or different segments of what we consider our seasonal climate, which might shift with climate change. So that is something that we may have to look to as an emerging threat. Now, the vast majority of these dengue cases we know were asymptomatic or mild, or which in some cases could be self-managed. However, it is still an alarming number, and we have to make note of this. We know that there are some cases of dengue which can essentially become extremely symptomatic, right? And if not managed carefully, can lead to hospital admissions, and if not managed appropriately, can potentially be fatal. We also had Zika and chikungunya, right? And the interesting thing about Zika and chikungunya, we say that they maintain similar transmission cycles. We know that both of them originated from Africa, but appear to have invaded the Western Hemisphere in the same year, right? The first outbreak of chikungunya in the Western Hemisphere occurred in the Caribbean island of St. Martin in 2013. Zika virus was detected in Brazil, but molecular clock studies have proven evidence that this virus was circulating undetected as early as 2013. There's so much that has been said about COVID and the virus that essentially would have led to the spread of that condition. We know that it is, well, we can say we know that it is, because we have not seen the last of COVID. It is still highly contagious, especially newer strains, more so than the ones which have been around for some time. And even in the space of vaccination, we still have to be mindful about the level of spread that could potentially arise from COVID. I know in some of the islands, we have been fairly good at containing the spread of COVID, but of course, we know there are pockets, etc., in different parts of the Caribbean and some of our countries where COVID still continues to be a problem, right? So we know that it had a very catastrophic effect around the world, especially in the year 2019 to 2020, resulting in more than 6 million deaths worldwide. After the first case of this predominantly respiratory virus illness was reported in Wuhan, China, in late December, we know that the disease spread right throughout the globe, and we had to deal with COVID within the Caribbean setting in a very low resource setting. So these cases that I've highlighted, they may not necessarily be new when I started my remarks, but we sense that without proper public health strategies, without proper education, sensitization, training of health personnel, and collaboration within the health team, right, or within the healthcare system, these agents will continue to be a problem for us, especially antimicrobial resistance. So I want to just say a little bit about AMR, because this is an area that, of course, is related to my field in pharmacology, because, you know, we're the ones that investigate the drugs, develop drugs, right? We're looking for new drugs from various sources, plants, natural sources, among other areas, right? But with AMR, a lot has to be said about, you know, this One Health approach that we have been talking about for some time. And I think we have done enough talking. We need to move into action because we have all the evidence which speaks to the potential cross-contamination between food and human, right? And if we are applying all the necessary techniques to safeguard human health, we should also be employing similar techniques, if not different techniques, to even monitor animal health, because there's a strong likelihood, right, of cross-contamination between animals, livestock, which essentially become food for most of us, apart from the vegetarians. But we should also speak about the potential cross-contamination between vegetable and food crop and humans as well, right? So it's not only animals and humans. But we know that antimicrobial resistance essentially generates when these microbial cells are sophisticated, as small as they are, they're extremely sophisticated, right? And they have developed sophisticated mechanisms to essentially ward off the effects of antibiotics. Hence, these medicines become less effective. And this has emerged as one of the leading public health threats of the 21st century. The review on antimicrobial resistance commissioned by the UK government and published in 2016 argued that AMR could kill potentially 10 million people by the year 2050. And some of you might be thinking that 2050 is a long way from now, right? But we can't just think about ourselves. We have to think about the generations to come, right? The One Health approach has been itemized as an area that we should place our emphasis and our focus, because that is where we potentially could help to curb cross-contamination between human and other potential hosts of microbes. So colleagues, as we consider charting the way forward, it is important for us to identify diagnostic and therapeutic measures which can mitigate the effects of these threats. And it is my hope, as Dean, Faculty of Medicine here at CAVIL, that your symposium and other symposia like yours, especially focused in this very specialized area of clinical microbiology, can help to stimulate thought, focus, research, and action as it relates to identifying solutions, which is encapsulated in your theme for this symposium. The Faculty of Medical Sciences is committed to this mission. And during the COVID-19 pandemic, we had faculty members, we had students that participated in vaccination programs and public sensitization, health awareness fora, and we will continue to do our part. In the area of AMR, the faculty has sought membership on the Food and Agriculture Organization of United Nations, AMR multi-stakeholder platform. And we see this as an opportunity to share with colleagues, the network, and to identify best practices, which we will not only pass on to our students, but also to the wider community surrounding the university. We know much more coordinated efforts are required to reduce disease burden of these pathogens that lead to these infectious conditions. And it is our hope in moving forward that these interventions will be able to be applied not only within the clinical setting, but as it relates to how we educate our young medical doctors and other healthcare professionals, right, as it relates to microbiology and the threats that could stem from microbes. Look at developing health policy. This is one of the areas that we are very passionate about. moving all of this research, into action and having the policies to support that action. And then last but not least, practice by providers, right? And the persons that work within the space, the professionals, and also our patients. So colleagues, thank you again for this opportunity to share these remarks. Hopefully I didn't carry on for too long. I will have to demit the meeting at some point because I have another meeting coming up, but I wish you all the best on your symposium, and I look forward to hearing about the outcomes of your meetings. Thank you.
And thank you, Dr. Kohal, for sharing those wonderful ideas with us this morning. You really have brought us up to date on the viral threats, on antibiotic resistance, and of course, the One Health agenda, which we should all get on board with. Thank you so much for taking time out of your schedule to provide opening remarks for our organization this morning. Thank you, thank you, thank you.
Thank you. All right, good.
And now we will continue our opening session with remarks from the president of the Caribbean Association of Clinical Microbiologists, Dr. Glendy Reynolds Campbell.
Good morning again, everyone. Apologies again, I had to shift my location because of internet challenges. I do hope you're not hearing all the ambient sounds around me. Good morning to everyone, everywhere that you are today. Thank you for joining us for our 19th annual symposium. As we've been mentioning, that big C, COVID, has brought on so many challenges for us. We were used to having our meetings face-to-face, and this is now our 4th year of having our meetings online. But all being said, it has made quite a few of us more technically inclined. So we still have things to give thanks for. We want to also say thank you for joining us, for making this meeting a success. Year after year, you have been here with us. Last year, we also had a wonderful meeting, and through that, we had persons giving us suggestions as to topics, etc. We had accumulated that into a nice little package, and we had our summer school sessions this year, which were awesome. We had some wonderful speakers. We learned quite a bit. And of course, we've provided the information on our YouTube page. So you're all invited to go ahead and view these lectures. They were very, very good, I must say. And just for some housekeeping, just remind you to keep your mics muted until it is time for the Q&A session. And we have quite a lineup for you today, so stay tuned for that. Remember to also complete the survey at the end of the conference. I just put it back in the chat for you. You can use either the QR code or the link that's provided to get onto that survey so that you can actually indicate whether or not you need your CMEs and have some instructions as to how to get those. So again, I want to say thank you. I know this is a big topic for today, and I do hope that you will enjoy everything that we have to offer you for today. Thank you.
Thank you, Dr. Glendy Reynolds Campbell, for those opening remarks. And now we're going to get right into our program for the morning. I would like at this time to introduce our first speaker for the morning, Dr. Marquita Gittins St. Hilaire is currently a senior lecturer in microbiology at the UWI Faculty of Medical Sciences, Cave Hill. She received her PhD in microbiology from the UWI Cave Hill in 2003. Her principal area of specialization is in molecular diagnostics of infectious diseases, particularly arboviruses and leptospirosis. However, she has a keen interest in the investigation and epidemiology of multi-drug resistant pathogens, in particular, the genotypic and phenotypic characteristics of these organisms. Dr. Hilaire is a member of a number of professional organizations relative to microbiology, and of particular, a member of the quadripartite technical working group on antimicrobial resistance and integrated surveillance. Colleagues, I am pleased to introduce to you this morning Dr. Gittins St. Hilaire, who will bring us up to date on emerging threats. What's next? Dr. Hilaire.
Good morning, everyone. I am going to share my screen now. So, good morning. I am Dr. Marquita Gittins St. Hilaire. As mentioned, I'm a senior lecturer in microbiology at the University of the West Indies, CACM. And we're going to talk today about what keeps emerging. We've been seeing in recent years, I mean, we're kind of used to dengue to some extent, having experienced some outbreaks from 1999 and moving forward. We are even currently in the midst, in some Caribbean countries, in the midst of a dengue outbreak. But in the interim, we've had threats of, as mentioned before, chikungunya. We've never seen that. We've seen Zika. We never saw that before. We even had threats of Ebola. And, various areas around the world, we are seeing threats of so many other infections. Of course, globally, there was COVID-19, which had us all on a standstill for a number of years. So we're going to look at what these emergent infections are to some extent. We're going to see why they've been emerging, what has been emerging, and then we're going to conclude on that. So in terms of these emerging infections, between 60 to 80% of all emergent infections are basically arriving from zoonotic, are basically zoonotic. They're derived from animal sources. And we have instances where this agent has been introduced into a new host population, as in one way, or the agent has been established and transmitted into a new host population. And we're seeing quite a bit of these when we look at any of the diseases that we would have mentioned just now, even moving into new host populations, the movement of Zika and chikungunya into our region. So the thing is that infections have been with us for thousands of years. And of course, because of the interaction between animals, between humans and their environments and our environments, we've basically had an increase and we've had changes over time in terms of these infections. So when we look at some of the reasons why we've been having these infections, why they continue to emerge, they have become, they've been emerging because of, once again, our changes in terms of interaction with the environment, with each other, and even with animals. And this can become very complex and multifactorial. And, at one point, we thought that these emerging and re-emerging infections had declined. But, we're now seeing adaptations in other places. And we are seeing microbes adapting and changing. As I always tell my students, if we look at the history of Neisseria gonorrhea, when penicillin was first discovered, in 1920, into operation in 1940s, we could have treated Neisseria gonorrhea, no problem, right? Now, when we look at treating Neisseria gonorrhea, we have to move away from the beta-lactams and now go, well, at least from penicillin in the beta-lactam groups, and now go into the cephalosporins, the third generation cephalosporins, because of the resistance that has developed over time. And now we're even seeing, that drug doesn't even work in some instances. So, we've actually seen instances where Neisseria gonorrhea has now to be treated, at least in this one instance where I saw, with carbapenems. And that's, very unusual. And that's where we're going. So organisms are adapting, they're changing. And then we have a lot of ecological changes. We are becoming more urbanized. Our agricultural practices are changing. We're moving now into areas that were forested before. So we're now coming into contact with organisms that we would not have had contact with before, or and hence the animals are coming into our territory. We have changes in irrigation. We have, nitrates and so forth being placed into the environment, changing our underground water system. We always also have climate change, which is a big thing because of changes in temperature, or rainfall, or changes in precipitation. We have organisms that are now arising in other places where they never were before. Dengue has always been associated with tropical and subtropical regions. Now we're seeing it now more so to some extent setting up itself in temperate regions. So, and we're having the organism is being able to spread among those populations because the vector is now able to survive in those areas. We have areas where flooding, because of climate change, we have flooding occurring more with greater frequency, and that increases our risk for waterborne infections, the leptospirosis, the schistosomiasis. We have all of those that can be associated with climate change. We also have human demographics and behavior. Changes in sexual behavior has increased the prevalence of STIs. And we've seen, if we look at some of the stats that have been put out of some of these European countries, we have increases in syphilis. Even if we think about monkeypox and its development or its rise to prominence even more so last year, you can see that it was in certain groups and so forth. So our changes in demographics or changes in behavior, this has led to the emergence of some of the organisms that we're seeing. The changes in technology and healthcare. We have certainly to think about globalization, traveling and industry. The fact that we can go from one part of the world to another part of the world in less than 24 hours means that we can take our organisms with us. If we think about COVID-19 in 2019, when it was first kind of described clinically in Wuhan, China, in the Caribbean, we weren't thinking about that, because I remember having some discussions about that, about COVID-19 and this particular new virus that's coming up, and you were thinking about bats and all the pangolins and all the other trade issues that have been associated with it. But then by March, probably around March 16th, 17th, 2020, we actually had our first case of COVID-19 in Barbados. And so, it didn't take very long. Before, years ago, it would take 2 months to get from one place to the next. It doesn't take that length of time anymore. We also have breakdown in public health measures, especially when we're thinking about wars and war-torn places where refugees are now seeking shelter in other places because of the close confines and so forth. They're able to now spread these infections. Infections can spread amongst themselves because the level of sanitation has broken down. And there's also greater susceptibility to infection when we think about other organisms such as HIV. And now that you have a greater influx or a greater increase in TB cases and persons having cancers and being immunocompromised or being on immunosuppressive therapy, it basically increases their risk. So even though we have greater longevity, it also to some extent increases our susceptibility to infection, particularly as our immune systems become a bit older. When we look at the timeline of these emerging infections, we can see that there have been and particularly just looking from 2003 to 2022, we can see that there have been a number of organisms and so forth that are being seen. We have Charpera virus that was seen in Bolivia in 2003. And that was a new virus in a new area. And then we have to think about chikungunya in Italy as well. Certainly a new virus, another virus in another area. So these are viruses that have been existing before, but now they've moved themselves into a new geographical location. And if we look closely at the Caribbean, we can think about cholera in Haiti in 2010, and that caused an outroar of its own. And then we think about chikungunya in the Caribbean in 2013, and Zika also in 2015 started out in Brazil at first, but then it also very quickly moved to the Caribbean. And the story of chikungunya is, I remember going to a conference in Jamaica in 2012, and we were talking about chikungunya. It just seemed like so far away. And by 2013, within about 12 months or less, we were seeing it circulating in the Caribbean, and of course, raising its head in Barbados. And then we've had other outbreaks of diseases as well. We think about COVID-19, SARS-CoV-2, which spread globally. So that's a totally new infection, and that was in 2019. And then we had re-emergence of monkeypox, which was also global. But then we had new infections in 2022, like the Lanya virus in China, which is a new virus that has now developed in China. Of course, looking at the geographical spread of these infections, globally, we have in 2009, we had the influenza virus, the H1N1. In 2019, we had SARS-CoV-2, but then we had a re-emergence of monkeypox in 2022. But then we've had new infections arising, or new organisms arising in different places of the world. So we have, like, if we go from North America down, we can see that there was the Heartland virus being seen, the influenza virus. So we had avian influenza virus, we had Oropouche virus as well in Suriname. We have the Charapé virus in Bolivia. And as we go on, we can see a number of these new viruses, these newly emerging infections in blue all over the world. There's a predominance of them circulating in China, and also in that Southeast Asian region, we're seeing an emergence of new infections as well. What we're also seeing in red is the new diseases that have moved into new areas. Of course, we can think about dengue, we can think about Zika. Even though dengue has been with us for a while, it wasn't always with us. But then we can think about chikungunya, which is more closer to us in terms of a new moving into a new area. So we can see that there have been circulations of these viruses over time, and they will continue to do so based on the reasons for the emergence. We're seeing, as mentioned before, instances of climate change that's happening all over. Before sometimes the temperatures in Barbados, I wonder if, we're going to cook. So yeah, we've had some significant changes, as we can see, in terms of climate change. And this, of course, affects the transmission of the viruses. So what infections are we going to be talking about? What's the what's, right? So WHO has basically developed a priority list of diseases that pose the greatest threat to public health because of their epidemic potential, or should I say sometimes even their pandemic potential, and whether there is no or insufficient countermeasures. So of course, one of the first things we need to think about is COVID-19, where it was not on our deck before, but it certainly has arisen. We have the Crimean Congo hemorrhagic fever, which has been in circulation in Africa, but now it's getting, particularly in Nigeria, but now you're seeing it circulating a little bit more, getting closer to home. We have Ebola, we have Marburg, that those viruses have also been endemic in Africa, some African countries, but we're seeing like Marburg has been moving to new places like Ghana, where it wasn't before. There's also Lassa virus, which has also been, we also think about as well. There's SARS, there's MERS, the Middle Eastern Respiratory Syndrome, and then there's Nipah virus. So these all generally are, they either have very rapid spread, they have a very high mortality rate in some instances, mortality and morbidity, and there are either very little measures to counteract them or there are insufficient measures to counteract them. So what are we going to talk about first? And of course, we're going to look at something that is a little bit further away from us, but it does have the potential to spread. Now, before the current outbreak in India, there were only 634 cases, and look at the amount of deaths. It's almost about, just past 50%. So we can see that this particular organism can result in significant mortality. And this was first identified. The first outbreak of Nipah was in Malaysia, Singapore between 1998 to 1999. And it was initially thought that it was a Japanese encephalitis. However, upon further investigation, they realized that it was the Nipah virus. And this is a BSL4 pathogen. And the major clinical manifestations in humans are respiratory and neurological signs. If we look a little bit at its virological characteristics, it's a negative single-stranded RNA virus belonging to the Paramyxoviridae, and it's also closely related to the hendroviruses. They're actually quite close. And the naming of the virus, the Nipah virus, was basically after the state in which it was found. The outbreak was found in the village of Songhai, Nipah, and so that's where it was first. That's where the name of the virus came from. And if we look at the map in terms of where it is located, we find that it is the predominance of this infection is found in the southern eastern countries. Malaysia, of course, where it was first isolated, Singapore, the Philippines, Bangladesh, India. So all in this area in yellow, and we see some of in the areas where you see the red, that's where they've had this Nipah virus infections. And in terms of where you would once again see the outbreaks and look at the numbers, you can see quite a bit of these infections have been occurring in Malaysia, of course, we will see. And then if we look at the mortality, we can see the mortality rate has been very high in India. So between 68 to 100%. In the Philippines is also pretty high as well. And then in other countries, we've seen a less degree of mortality. But in terms of the infection, we have for right now, is confined to this area, but the mortality rate is quite high. So what is responsible for this? So the Nipah virus infections have been linked to the geographical areas that carry a specific bat, known as the frugivorous bats. They're called flying foxes, and they belong to the Therapus genus. And where do we find these bats? Well, in the same places where you find the infections: Malaysia, Singapore, Bangladesh, India, and also the Philippines. And where the infections have emerged is where not only where the bats are, but where they've been removing the bat habitats. So where the bat habitats have been lost is where you're seeing the infections occurring. So the bats are the main reservoir for these Nipah viruses, and they're able to cause spontaneous disease in humans, in pigs, in horses, in cats, in dogs, even though the natural hosts for these are the fruit bats that we see here. hanging from the branch. So these are primarily involved in the spread with direct shedding to receptive animals, humans being included, and they can also be through the contaminated palm fruits or the palm sap, or via the urine or feces that is produced by the bat. They've also been seen in pigs, as I mentioned before, horses, domestic and feral cats. And the transmission can be intraspecific, so it can go from pigs to pigs, humans to humans, or it can be intraspecific, so it can be from a bat to a human, from a pig to a human, from a horse to a human. And they've also seen it in other animals as well. It can be seen in peridomestic and feral cats, but these are not known to be a reservoir, and the transmission risk is quite low. So now we have a fight on our hands in terms of, the virus versus man. At some points, it seems as though the virus is winning out, because at the end of the day, the majority of persons who acquire this infection, they do not survive. And as mentioned before, the majority of the infections are either CNS related. So the persons will present with encephalitis or some form of brainstem dysfunction or seizures or even meningitis. And then they can also present with psychiatric symptoms, but we can also have respiratory tract infections as well. When we look at the differential diagnosis, I picked these ones because they're more closer to home for us. We can think about cerebral malaria, we can think about leptospirosis and some of the presentations. And a lot of these viral infections tend to present very similarly, particularly in the early stage. So the dengue encephalitis, herpes meningoencephalitis, and because of the travel that occurs globally, we are able to have transmission of these infections elsewhere. We also need to include on this list bacterial meningitis and rabies as well. In terms of diagnosis and treatment, we can do routine tests, but they're very specific tests for them. For the virus, we can do PCR on some of the samples, urine, the blood, and we can also do virus isolation techniques. Most of the treatment, so there's no specific treatment for it, mainly supportive and syndromic management of the acute encephalitis syndrome. There's 3 pharmacological treatments that can be applied, which is ribavirin. There's a monoclonal antibody, and favipiravir, which is a antiviral agent that can be used. But as mentioned very nicely by Dr. Kohal, is this One Health approach. That's the way in which we can certainly counteract this, and we certainly have to start now. So if effective control measures in the community to prevent the transmission from animals, bats and pigs to humans has to be instituted and have been instituted to control the spread of this particular infection. We also need to, going on to another organism. In 2017, the World Health Organization published a list of priority pathogens, which included 12 families of bacteria that pose the greatest threat to human health. So the major objective of this was to prioritize funding and incentives to align research and development priorities of public health relevance, and to garner global coordination in the fight against antibiotic-resistant bacteria. And you would have heard some very sobering statistics being given by Dr. Kohal as well. And we've had a significant burden of this, of antimicrobial resistance. It's been said that they estimate that about 4.9 million deaths were associated with bacterial antimicrobial resistance in 2019, including 1.27 million deaths attributed directly to bacterial AMR. And the organisms that cause the greatest havoc are some on this list. We have Staphylococcus aureus, Klebsiella, pneumoniae, Streptococcus pneumoniae, Acinetobacter baumannii, Pseudomonas aeruginosa. These have been responsible for nearly 1 million deaths, and at least more so as time goes forward. So you can see how they have been listed. When we look at Acinetobacter, so we focus on Acinetobacter baumannii. So there have been a number of organisms that have developed resistance. As I said, we look at Acinetobacter baumannii, but we can also look at Pseudomonas, which causes also a lot of havoc as well. And we're going to focus primarily on carbapenem-resistant Acinetobacter baumannii. This organism, we know, is a gram-negative, non-fermentative, oxidase-positive aerobic coccobacillus that is found ubiquitously in nature, and it can also be found very prevalent in the hospital environment and has been associated with a variety of opportunistic hospital-based infections. We can isolate this organism from soil, from water, from the environment, and from skin. And the thing about it is that it has been associated with a lot of infections in the hospital. So it's one of the most commonly occurring infections in the healthcare environment, particularly for persons who have underlying comorbidities, who have invasive devices in place, and who received an extended course of antibiotics. And now it's recognized as one of the world's leading causes of healthcare-associated infections. And it has a very high mortality rate. It also has unfavorable clinical outcomes. So the carbapenem resistance has been acquired through a number of mechanisms, some transmissible genetic elements, and the carbapenemases, the enzymes that render the carbapenem ineffective, have been seen for a long time. And we down here, we have KPC, which we all are familiar with, the Klebsiella pneumoniae carbapenemase, but then we have some more down here as well. And then we have the OXA ones. So the ones at the top tend to be the ones that are most common, and the ones at the bottom, the OXA 23, 24, 40, and 58, like oxacillinases, tend to be less common. So these resistant organisms are the carbapenem-resistant Enterobacteriaceae. The organism we're going to look at, as I said before, is Acinetobacter baumannii. These are generally broadly resistant to all the carbapenems: meropenem, imipenem, doripenem. And the beta-lactamase that the organism produces is also effective against third-generation cephalosporins. And so you can also see other types of enzymes being produced, such as the C beta-lactamase, and those that are specific to the third-generation cephalosporins would be your extended spectrum beta-lactams. But when combined with porine mutations, can also result in the carbapenem antibiotics from entering the cell. So on the first carbapenems that was identified was the Klebsiella pneumoniae carbapenemase in the United States, and then this has subsequently disseminated all across the world. And then we have the OXA ones, OXA 48 like that was isolated also from Klebsiella pneumoniae in 2004. All of these, as you see here on this table, the carbapenem-resistant Enterobacteriaceae, all of these carbapenemases, along with some non-carbapenemase mechanisms like efflux pumps, are able to work effectively at getting the rendering the organism resistant. So we have porine sizes can change. We have a number of efflux pumps here at the top, and we can have change, and they can result in resistance to a number, not only the carbapenems at the end of the day. That's what we're talking about, but they can also render resistance to drugs such as the fluoroquinolones. And so what I'm saying at the end of the day is that it makes it very, very difficult for us to treat this organism. And so if we look at the timeline for this particular infection or this particular mechanism, we can see that over the years, we've developed, or numerous carbapenemases have been developed in various countries, and they've been spreading all over the world. So in 2022, just last year, the Infectious Disease Society of America and ECCMID, they kind of developed a protocol for treating carbapenem-resistant infections. And they basically came up with the same type of treatment options. You can use, depending on whether it's mild or moderate, you can see that you can have various agents that can be used to treat the infection. You can use ampicillin sulbactam as the preferred agent for mild or polymyxin B, and sidofuricol can also be used as well, but it, is used in particular cases. And this is basically not just to go through and show you what can be used to treat it, but to show that it's been on the radar, and they're looking at ways in which it can be treated appropriately. And across the world, particularly in these two organizations, one in the East, one in the West, they're basically working to make sure that there is a way to deal with this particular organism. And so they've also developed mechanisms for treating the same carbapenem-resistant Acinetobacter baumannii in the ICUs. They've developed specific strategies and so forth for treating it, for controlling the use of carbapenems, because obviously in ICU units, what happens is that what happens in ICU units, because of the severity of the infections and the prognosis, the poor prognosis, you generally have to start with a very broad spectrum antibiotic. And therefore you have to think about being able to use the carbapenems, which will be your very broad spectrum antibiotics, how to use them appropriately to manage these infections and to effectively, and also in curbing or curtailing or modifying their use, so that there can be incidence of carbapenem resistance in ICUs can be reduced. And of course, there are things like early diagnosis, early screening, looking at fecal carriage in the spread of it, looking at early detection methods such as LAMP and whole genome sequencing as well. And then there are other interventions such as the infection control measures and the measures that prevent spread, such as bathing in chlorhexidine or so forth, that would reduce the risk, reduce the use of ventilators, make sure that the area is appropriately clean to prevent transmission. And finally, we'll look at our very last organism, and we'll look at fungi. So only this year did the WHO actually create a list of critical fungi, a critical fungus list. It wasn't in existence before. So we know that we looked at the bacteria, but we never used to look at the fungi. And the burden of this was never estimated because, it caused serious and life-threatening infections, but was all overlooked for bacteria. So the fungus in particular have been largely neglected, despite the costs incurred in the healthcare due to antifungal resistant organisms that have been exemplified by the emergence of the global spread of multi-drug resistant organisms such as Candida auris and azole-resistant Aspergillus fumigatus. And so in this context, the WHO launched the fungal priority pathogens list and also similarly ranked them, like how they did with the bacteria, into critical, where we have Cryptococcus neoformans, Candida auris, and the same Aspergillus fumigatus and Candida albicans. We have the medium group, and we also have the high group, because these infections have been found particularly so in immunocompromised patients. With Candida auris infection, it belongs to the genus Candida, and it's been named because it was first found in the external ear canal in a patient in Japan. And its genetic profile showed that it was very close to some Candida species. And there are 4 geographical clades, one in South Asia, East Asia, South Africa, South America, and there's a possible 5th one in Iran. And the thing about Candida auris is that, it is, as we mentioned, a very multi-drug resistant organism. It's considered an emerging fungal pathogen because it's been primarily associated with hospital infections, and it is considered a serious global threat. It was first observed in 2019, and now has been isolated in 35 countries except Antarctica, and it has a very, very high mortality rate. When we look at how it is transmitted, it's usually done so via person-to-person spread, which is unlike how other Candida species are acquired. So Candida auris is not considered a resident commensal organism and is not typically present in the human gastrointestinal tract, as are many of the other Candida species. It also has a predilection for certain areas of the skin, such as the axilla and the groin, and it also has the potential to colonize hosts within days to weeks of exposure. And it can persist for months and possibly indefinitely. So it can spread via fomites and contaminated surfaces, and it can remain viable for 4 weeks and is culturable for at least 2 weeks. So we can see, look at this picture, that it can be found just about everywhere. It's found, has been found in patients' rooms, hallways, chairs, on the machinery that you use, on ventilators. It has been found, and because of its ability to survive in these dry or moist surfaces for 7 days, and also the ability for it to form biofilms, it means it can last there for a long time. So who is at risk? And we can see a long list of persons here who's at risk for this. And we know persons who are immunocompromised, of course, because that's the general thing. But persons who are diabetic, persons who have chronic kidney disease, persons who've been on antibiotics for a long time, and any persons with any devices, those are also at increased risk. So once it's colonized, once colonization has set in, then it can remain on, as I said, these surfaces, these catheters and so forth, and then cause a wide range of infections, as you see here on the right. So it can affect the bone, it can get in the GI tract, and can cause urinary tract infections, it can affect the heart, it can affect the eyes, it can cause a wide range of infections. So in terms of resistance and treatment, the CDC has reported that 90% of these isolates are resistant, and that's in the US, are resistant to fluconazole, 30% resistant to amphotericin B, and 5% resistant are resistant to the echinocandins. And we look at treatment options, we don't have any triazoles or azoles in here for treatment. Most of them are the echinocandins. So they are what are used primarily for treatment of Candida auris infections. Of course, we can have combinations as well. So at the end of it, we have, a widespread international travel, tourism, population changes, migration, changes in demographics, patient transfer, the association between a specific resistance mechanism and a given country or region may change. And this would create an urgent need for routine local, national, and global surveillance mechanisms for carbapenemases. But this is not only for carbapenemases, it's for all those that I mentioned before. So it's not only for resistant bacteria, but it's the same for viruses. It's the same for fungi. They all need active surveillance locally, nationally, and globally. And also you need to include the research and development for drugs that can be used to treat them. And of course, I need to put in the plug for the One Health mechanism, particularly to reduce the incidence for antimicrobial stewardship as well, to ensure that the drugs are used appropriately, and for infection control as well. So I just want to thank you for taking your time, and thank you for your attention. And if you have any questions, any time remaining.
Okay, thank you so much, Dr. Gittens St. Hilaire, for this very comprehensive update on emerging threats. It was truly enjoyable to learn so much about what we'll be facing in the future. Thank you so much. And at this time, we will entertain any questions. Please raise your hand if you have any questions for Dr. Gittens Hilaire. Dr. Reynolds, can you help me see who answered?
Yes, there are some questions in the chat. We're going to ask Dr. Gittens to just stop sharing her screen at this time. Okay, so Ariane is asking briefly, what do current carbopetimesis surveillance programs look like in the Caribbean?
Well, a lot of, in terms of the Caribbean, I can't speak for the entire Caribbean as to what, is happening. But on the ground, they're always monitoring the incidence of carbopetimesis and the occurrence of carbopetimesis in Barbados. That has always been something that's been done by the hospital. Even in terms of when they have outbreaks, it's always been monitored. And so the appropriate measures have always been put in place. But internationally, there are some mechanisms such as GLASS and so forth that have been produced by the WHO and the quadripartite that have been used to acquire the data from the other from all the countries in general and be able to compile this data to see what is happening within the respective countries and then to obtain a global picture of what's happening. So it would be good. I am currently a part of the quadripartite technical working group on antimicrobial resistance, use, and integrated surveillance. And the whole thing now is just to find a way in which you can use integrated surveillance or how you can perform surveillance within the respective countries, and then how to bring it on a grander scale so that we would know exactly what's happening. Of course, as I said, there are mechanisms like GLASS and so forth that have been produced, but what can happen within each country to be able to see what's happening in that country, and then to have a cumulative look at what's happening globally and how to counteract it then.
Okay, thank you for that. I don't think we have any more questions at this time. Anybody else would like to put a question in the chat or raise your hand? All right, so we're moving right along. You're muted, Doc. Dr. Lewis, we're not hearing you. You're muted.
I am muted. Okay, we're going to move right along. Our next speaker is Dr. Edmund Blades. Dr. Edmund Blades is currently a non-clinical consultant microbiologist at the Queen Elizabeth Hospital. Dr. Blades spent many years as a medical technologist, specializing in the disciplines of hematology and microbiology. He has always had a very keen interest in research in microbiology, and this led him to pursuing the PhD degree at the University of the West Indies Cave Hill campus, which was awarded in 2014. His research was primarily in the area of aerobiology, where he studied the microbial flora of the African Sahara dust. This led him to becoming a member of the Pan American Association of Aerobiologists and also a certified mold inspector and spore analyst. Dr. Blades spent many years working in public health and was awarded the Barbados Service Star National Honor for his years of service in public health microbiology. His current interests include indoor environmental air quality, antibiotic stewardship and resistance, and he enjoys working with the linkages to public health issues and the One Health agenda. Colleagues, I now present Dr. Blades, who will bring us up to date on antimicrobial resistance in a primary care institution in Barbados.
Good morning, and thank you, Dr. Lewis, for the very kind words of introduction. I thank you all for the opportunity to present this morning. I really enjoyed so far the proceedings, especially Dr. Giddens' talk about all these microbes and these new diseases coming in hand. So they want to make a contribution, talk about an update on AMR in a primary care institution in Barbados, which of course happens to be the Queen Elizabeth Hospital. I'm very careful as to how I present this data because much of it is not yet public and I want to be discreet in how much I give today. So the background to all this is that AMR, as we know, is described nowadays as a silent pandemic. And there are many, many efforts on many fronts to try to curb the spread, including all the way up to the WHO and to the Global Leaders Group. And we know that our Prime Minister Barbados is co-chair of that group. And therefore, Barbados has, I suppose, a more vested interest in making sure that the Barbados picture is well documented and efforts are made all the time to keep abreast of what's happening at our institution. Just last Tuesday, our Minister of Health, in addressing the Caribbean Association of Medical Technology and AMP, we made a point about future-proofing the laboratory. And in that address, he talked a lot about AMR. And therefore, the AMR business approach pervades the whole top level of the Ministry of Health, and it trickles down to all of us in the laboratory. And there's so many stakeholders involved in making sure that we in Barbados do as best a job as we can when it comes to monitoring AMR. We've had our challenges, of course, but our efforts are continuing to do our best. I want to show this picture quickly, and the last part of the slide at the bottom of the writing talks about AMR being an extremely important matter for us all as it threatens global health, food security, and sustainable development. These are terms that we come to grips with, that AMR, if not unchecked, can have a very pervading effect through the whole society. And therefore, we must work together, not only as global leaders, but as all involved in the healthcare industry to make sure we have hands-on, so to speak, on AMR. So this group, really seeks to give those on the ground, bring awareness, advocacy, and try to help us provide funding for our equipment. We can't do AMR properly except we have the correct tools to do the surveillance and the identification and the follow-up. And therefore, equipment maintenance acquisition is critical. And this is one of the areas that Barbados has to try its best to keep on the cutting edge so as to capitalize on the gains, so to speak, we've made so far. So what's the strategy and the progress in Barbados in terms of AMR surveillance and monitoring? We have a national action plan on combating AMR 2021-2026. And this is really the broad scope, the broad framework of activities that lead up to the monitoring and so on of AMR. And this is a national effort, and there are several groups on this committee. There's several tripartite groups. We heard Dr. Giddens talk about them. And it's really a challenge and a privilege too, to be able to be involved in this kind of work in terms of monitoring and seeking to put our efforts here locally on the map. So this is, of course, led by the most honorable Dr. Corey Ford, but there are lab people involved. There are the medical staff involved. There's staff from the pharmacy, staff from the MICUs and so on. And as we progress in this forum, we're trying to improve and involve other areas beside the hospital so that there's a total overall picture of AMR when it comes to public health, when it comes to human health. I think at some point in time, others will speak on the animal health, but for us, we're concerned primarily about human health. So one area that we're one now is the outpatient clinics. We try to monitor the hospital inpatient, but what is happening to those on the outside? Are the antibiotic patterns the same? Are we having the same bacteria and bugs? Are we having a different response to treatment? Are we having the same focused ideas on treatment? And that has to be reined in and guided so that we have a combined effort and approach. If not, on one hand, you make some progress, on the other hand, you make a step or two backwards. So it has to be a focused approach. It takes time, it takes effort, it takes dedication. So in our scheme of things, what happens is that as the hospital laboratory identifies especially resistant bugs, especially the enterobacteriales, and those kind of organisms, the gram-negatives, we flag them. Our instrument is the autoscan, and we can pick up the resistance bugs on that profile of antibiotics. When we see, and this has broadened the scope a lot, when we see the resistant carbapenems, that's perhaps one of our biggest flags to work on further on identifying that bacteria as well as the resistance mechanisms. Why is it resistant? And you just don't want to leave it and say it's resistant. But as we progress in our efforts, we want to identify the resistance genes. We want to find opportunities and the materials so that we can come to grips with what is actually circulating in Barbados. And as this is a work in progress, we now have an arrangement where we send all of these resistant isolates to the best standards public health laboratory. So the Barbados model is that all clinical labs in Barbados, both public and private, are to submit the resistant isolates to the best standards public health lab, where they will then be characterized further to develop the resistance mechanisms. So we know that some labs are challenged in terms of the equipment, the training, the reagents to do the detailed work. And therefore, the public health lab has been tasked with that job, and there have been several training opportunities for staff at the Best Science Lab to be trained in Argentina and elsewhere to have the knowledge and the expertise and the equipment and the reagents so that this lab is now focusing on gathering all of the resistant bugs in one place to do the characterization and to finally come up with a pattern so that we can have a national picture of what's happening. And this has been a work in progress. We understand that some labs are more challenged than others. We understand that the process will involve a lot of staff time and so on, but gradually we've come to grips with the fact that this is our job. And we have to make a bigger effort in trying to identify and categorize our resistance problems in Barbados. And therefore the QA, I suppose, is the single most institution that will have a reservoir of these bugs. There are patients in and outpatients, and we have a ready capture area for these bugs. And the lab staff will have additional responsibilities to make sure that this kind of surveillance work in terms of gathering these specimens, making sure that they are properly categorized, making sure that they are sent over to the Best Science Lab, and this is a work in progress. And then recently, in order for us to really analyze the data, there's the HUNET software, which is a free software available on the net that has been offered to us. I've been doing the antibiogram for the last couple of years, but recently in September this year, there were 28 persons trained from all three sectors, both human, animal, and environment, in the use of and the proficiency of using of this software. It really makes life easy where you can just input the data from your instrument, whether it be the autoscan or whatever it is, that can then use that raw data to generate for you all your stats you need and generate an antibiogram. So the QH antibiogram that has been produced is being used by the doctors, by the pharmacists, and so on. And it really is putting together all the data that you have from different areas, and we can do a generalized antibiogram for the whole institution. We can then pull out from that antibiogram certain target areas, the ICUs, the emergency department. We can do the blood cultures, we can do the urine, we can do anything that you want to look at to see trends. Again, this is a work in progress, but we are heartened with the fact that there's been training and that more people are able to use the software. More people have more confidence in using software. And I'm hoping that as a result of this training, that there will be more input and therefore more outputs from this work. We continue to monitor outbreak scenarios. When we see trends upwards, especially in bad bugs, we want to do a categorization and do an investigation. We had two sorts of outbreaks of Burkholderia in 2018, 2023. And Burkholderia is a very water-loving environment from water sources. And this can find its way into the hospital, into patients, especially into blood cultures. And that was the case 2018, 2023. I'll show you later on. And then we're noticing from the antibiogram a rise in antibiotic resistance. Is this real? What is the cause? How do we define it? What do we do to stop it? We have to be aware first. We have to look at the data. So data management is so critical. We have the data, we can then do lots of things with data, but we must have clean, good data. And we're not perfect yet, but as we progress and we learn, become more proficient at these things. And then finally, what do we identify the resistance genes? Do we have endemic coli? Do we have, MBLs? What is the driving factor behind the resistance, if any? So the antibiogram, according to the CSI standards, is really an overall profile of an antimicrobial susceptibility results of a microbial species to a battery, a whole set of antibiotic agents, which will be used in patient care. And this must be used in conjunction with the institution's formulary. So you don't just get the antibiogram and put it on the table. What are the available antibiotics nationally that can be used? There's some antibiotics that are not available in Barbados, but they're available internationally. So what do we do with the data? We have to be able to put it in such a way that we can use it locally, and therefore we have to have the right antibiotics. Does it make sense reporting antibiotics that we don't have or can't get because they don't help to us? The antibiogram is a local antibiogram designed to be used in a local place. Therefore, we have to have the correct antibiotic and access to these things. So the antibiogram is used as an empirical guide. In other words, if a doctor suspects an infection, it takes a sample, but it cannot wait on a routine sample being done routinely to take 48 hours, start treatment. Therefore, he looks at an antibiogram and he suspects that this is a urinary tract infection. What is the most common organism from urines? What is the most usable, likely to work antibiotic in urines? If you have a historical view from an antibiogram, he's able to choose an antibiotic that is most likely to work. If it doesn't work, then he has a report in 48, 72 hours that will tell him specifically what the antibiotic is. Until then, he may use the antibiogram to guide him empirically to make decisions that are based on his institution's guide and historical evidence that we have these things in certain patient areas that will work. So if you may have a different profile, you may have one in A&E, but you have these targeted and guided antibiograms to guide him or her in the initial treatment when the patient comes in to his office. I recently had a call from a doctor saying he had not had a report for 7 days because in the laboratory, people don't always understand that cultures have become mixed. And when you have mixed cultures and you put a mixed culture on an instrument, the instrument can't tell it's mixed except it gives you garbled reports. And sometimes you get reports that doesn't make sense and you suspect that it's mixed. You go back to a plate which is pure at 24 hours, and at 40 hours, you see that there's something under it. And hey, that's a mixture. Then you have to go back now and sub again in that 24 hours. So it takes time, so it's a long time to get a report to a physician that will give him a direct result of an antibiotic. And some patients have 2 or 3 different organisms in the same sample. And therefore, it is a challenge to do that. All right, so here is a snapshot of an antibiogram for the QEH. It's kind of busy. I won't really go through all of it. But our top organism is in the top line. So this is the gram-negative bacteria. And on the top line, you have all the antibiotics that we use. And they represent different groups. So you have the cephalosporins, the first, the second, and third generations. You have other antibiotics. So the E. coli is our topmost isolate because, again, it's from the urine. The urine is our biggest, our most sample. And therefore, that will drive the antibiogram. And therefore, you can pull out a urine antibiogram by itself to tell you what's happening in the urine. And that may be different from urine from other E. coli from other places. So I want to highlight this column in red, the antibiotic literacy. We have a whole list of the most common ones, and the antibiogram selects must be analyzed on at least 30 isolates. So you have enough power to give you a viable result. So you don't want to take one sample or one isolate and do an antibiogram. At least 30 samples, that's the definition. So we have from 788, the E. coli, then the 30 for the Serratia. We have here Dr. Giddens, 47 Ebomanii in this list, and you have the profile there. But you have fully ranged Klebsiella pneumoniae, Pseudomonas, Proteus, Morganella, etc. So they're all there. These are all the common organisms, the most frequently identified organisms from the antibiogram. But I've been looking at the Enterobacter cholacea because there seems to be something brewing in that group. I'll show you later on what I mean, but I've seen a drop in the resistance patterns over time, beginning from 2016, 17, 18. I'll show you shortly. And this is how the antibiogram can be used. If you have a yearly picture of what you have for resistance patterns, and you look and you see that it's dropping or rising, then you can understand that there's something happening in the system. And once the data is a common pool, you can then begin to elucidate what the matter is and check and see what's happening. So Ebomanii is, yes, a very resistant organism. It has a lot of intrinsic resistance, ampicillins, augmentins, and so on. All these first generation cephalosporins, and you won't use those drugs at all. So the R's means that it's intrinsically resistant. Even if you get a pattern that's sensitive, you will not use those antibiotics because the bacteria has an intrinsic resistance to these things. So don't use those R's, right? The ones in red I've highlighted because I think that they're changing, and I will want to look at those later on, but those are the ones that we're looking at now. And you may have groups. You have the Serratia, which was 30 this year. There's no Burkholderia on this list because it was less this year. But for the total, by the end of the year, you will probably find there more than 30. This is the gram-negative ones. You also have the gram-positive one as well. All the Staph aureus, Endococcus, and so on. And I want to share here, Barbados has a peculiar situation with the MRSAs. Our MRSA rate is very high compared to Jamaica and Trinidad. I don't know why I've raised this question before. I'm just trying to find out why. We find that we have a 45% of staff OS, MRSA, sorry, among staff OS. And Dr. Giddens may want to comment on that later on. But MRSA is high in Barbados. And it's dropping now. It was high before, but it was at 57% before, now at 45% this year. So I think that as we look at this data, the powers that be, Dr. Ford and company can use targeted approaches to find out why this is happening, and then use that data to try to reduce the levels and incidence at MRSA, and for that case, any bacteria. So here we have the antibiogram, the gram-negative and gram-positive, and we can use that for several reasons and options to help us guide our AMR program. This is an old slide. I showed it before, but I want to make the point here, right? And this has not changed up in 2022. E. coli being the biggest, most populous isolate from urines. The antibiogram here shows all these drugs, antibiotics, which have various sensitivities. Nowadays, the trend is to use ciprofloxacin as the drug of choice. But if you look at the Bactrim has a little lower sensitivity rate than Cipro. But look at Nitro, 98%. Some doctors say nitrofuran is not an antibiotic and they don't use it. But it's an old drug which works. So when we look at the program to de-escalate and escalate things, you may want to start with an antibiotic that has less side effects, less problems, like nitrofuran, before you go to Cipro or one of the other heavier drugs. And this is the point. You use the antibiogram to get data to guide your empirical treatment. And if you do that, if it doesn't work at 24 hours, 40 hours, when you do a culture, then you can escalate up to a high antibiotic. But you should not really use the top heavy stuff first before trying a one that you know will work early on. So this chart here shows the Burkhardt outbreak in 2018. First point I want to make is that from February, March, the first bar go across, you see that there's some months with no Burkhardt. So it's not a common isolate. Some months you get 2, some months you get 3, the majority of months you get none. But in between March and July, so you had a whole heap, 5 is a whole heap in terms of one peak. But you had a 2, 3, 5, about 10 samples. This is a very rare organism. And you have to know when to call it an outbreak. Dr. Ford called it 1. So because they had this sudden increase and prolonged increase over a couple months, we kept recovering from blood cultures. This is blood cultures, these Burks, and therefore we call it an outbreak. And we can use this data from the antibiogram to retrospectively look back, see what happened. But the constant daily monitoring of the department has to be on top so that you can catch a trend. So when technologists see a bird today, 1 tomorrow, 1 next day, something snaps in the mind. This is not common. You see 2 on one day, 3 on one day. This is real time. You can then report to the FPC panel that we're having, look, something's happening here. Then they will go and investigate and find the real problem. This chart here is after the fact, showing the data, but you can do it in real time and monitor what's happening. This is 2023. We had a bigger outbreak, at least 15 samples in June alone. So you can use the data from the antibiogram to pinpoint and show you that there are trends, and you can use those trends to go back and see what's happening. In real time, this caused a huge spin-off of work for the laboratory, where as the investigation proceeded, you took several samples from different sources. All the swabs in the wards, you did the water And we actually found that the source was in water. And this is how the IPC team works, how we collaborate together, staff in the lab, staff in the ward, IPC, and we try to pinpoint where the problem is and make measures to correct it so that it doesn't happen again. Some bacteria are so, I can't say design, but they are prone to cause outbreaks. And this is one of them, as well as Serratia. So any bacteria can cause an outbreak. We have to monitor. So again, the point here we're making about the referral to the best standards lab is that training was provided by PAHO, others, and that we're now progressing to even more training, more equipment, and more capacity to find out through PCR and other methods what the real gene is, what's driving, what's the reason for the increased resistance and accuracy. That's the next project. But sorry, if you want to make this lab a regional reference point for the Eastern Caribbean, so that there's something in the country that we can send other samples from other labs to Barbados, so that there's capacity in the region to quickly respond to this kind of outbreaks. And this would involve those labs having proficiency testing so that they can become accustomed to identifying and recovering these resistant bugs. It's a work in progress, it's a good work, and I'm happy that Barbados and the division has something in-house, so to speak, in the country, so to speak, nearby, they can use as a referral lab. So, excuse me, from the blood cultures, the most common isolate in 2022 was Staph aureus. And we had an issue with that, not with that, but with what in 2018-16, our most common isolate was Staph epi. And I hope that slide is here, I can't remember, but we try to our best to look, because epi in the blood culture at more than 24 hours, it's probably mostly a contaminant. And we had to put measures in place. We use the data here to put measures in place and speak to the doctors and the clinicians about sample taking. Because epi is a skin flora, and if you get it in the blood culture all the time, it suggests that you're not taking your samples properly. And you're going to cause this uptick in bacteria that are not clinically significant. Not saying that all of them are not, but if you have this rise, this constantly high recovery rate of Staph epi after 40 hours, then the literature suggests that it is not a problem, that it is a thing. So here we have 105 in 2022. 2016, it was much, much more than that. It's higher than Staph aureus, higher than Klebsiella. So we talked earlier about enterobacter clavaceae. From the sources, urine, blood, joint, etc., we had 53. From the urines, we had blood 30, had bone, joint, itch tissue 24, bone 17, sputum 2, total 126. So this was what I pulled out from the data to look at. So this is the presentation there. Here's the trend over time, 2017, 2022. There's this rise from about 75 to 120 per antibiogram. And this is the first isolate only, not every isolate. So there seems to be a rise in clavaceae. This is what we can use the antibiogram for, to look for trends and follow trends. Now this is the antibiotic ceftazidime, which is a third generation cephalosporin. And you see here a drop in sensitivity. You know that was sensitive in 2017 was 85, 80 something. 2022 is down to about 60 something. So there seems to be an increase in resistance in third generations for equiracy. Now this may not be anything, but as you look at the data, you can pull these things from data. Here we're seeing this picture, and we want to investigate what's the cause. Blood cultures being a very critical isolate. I put out all of the blood cultures for 3 or 4 areas that are critically important: SIUs, the SICU, sorry, the medical intensive cares, and A&E. A&E gives us the most samples for any unit, but you see here that we have the big 3 or 4, pneumoniae, Clarissa again. The pneumonias are 14, 7, and 25. So that's the top one from the blood cultures. And you have the E. coli's and so the Monas aeruginosa, Staph aureus, and so on. And so again, the Clarissa is 5, 4, and 5. Is that something? You have to put that together with the other facts to look at it. Nowadays, we want to find an answer quickly. You no longer want to wait 40 hours for a blood culture report and sensitivity. We have on hand this BioFire molecular instrument, which can give you the ID of the bacteria and if there are certain resistance genes involved in it. It's a very quick test. You take a positive blood culture or any fluid, and you put it on the instrument with 2 minutes hands-on time to set it up, and you get a result in about an hour. This is very, very good. It's a very important tool, and we have a protocol for it. We haven't got enough for everybody. It's a very expensive test, but we want to gradually introduce it to the hospital, and we're targeting first of all the ICUs and any critical patients, which the doctor on the ward, the consultant, or Dr. Ford or myself can flag as critical. We don't see patients, we see samples. We haven't got the final say on who's more sick than who. But when they combine our efforts, we can come up with the fact that this patient is critical and we need to have a fast ID because his prognosis is not good. We suspect yeast, we suspect something else other than staph epi, and therefore you want to know quickly as possible what it is and if there are any resistance genes, so we can start to use something else other than the regular profile for antibiotic for blood cultures. It's a fast test. It will have benefits reducing hospital length of stay, patient survival rates, etc. And we want to have this technology introduced and running in Barbados. It's here. So, final slides. This was a challenge for some of us. We have to understand that all of the old bacteria are beginning to show signs of resistance. And many people are not yet attuned to the fact that things that were before not common are happening. This organism here is Shibata fungi, and it was on this form Hod's positive. And somebody said that's not possible. Hod is for Klebsiella. Hod is for this thing. Hod is to look for carpenter resistance. But here is a Shibata fungi. It's Hod's positive. Our protocol is to tell us, send over to pesticide lab. So we have on the form PHL, which is made. So this is a typical case of, this is a urinary blood culture. But if it was a blood culture, it would have gone straight on the BioFire. Because of the fact that we were querying and debating and discussing, we put it on the BioFire, and we got that it was Enterobacteriales because it doesn't have the name of Enterobacteriales in the bank. But of note is that it says here on the 4th line on top, resistance gene detected NDM. And that was a big thing for us. And then it says there, a tick detected NDM. So here we are having technology that we can get a result in an hour after a positive culture to put on the instrument. tell us what the organism is and what not to treat with, so we can have a better outcome for patient and better confidence in the results. So, the new kid in the block is these metallo-beta-lactamase mechanisms. And I have here a chart which we have from, which shows the results we're getting from confirmation tests from the Best Science Lab. And this is really good that we can do this. And I want to congratulate all involved with this work and to say that we're very appreciative of the fact that we can have this kind of technology. So one Pseudomonas, we have 3 Clavicii and one with both MBL and ASBL. 1 E. coli, and then 6 E. coli, sorry, 2 Klebsiella pneumoniae, one with both MBL and BSBL. And this Stitch-Barr frontier here, which is the NDM MBL. So that's new for us. And it shows that our combined efforts at the antimicrobial AMR surveillance is working. Once we have the focus maintained and kept, you will get good results for our patients. I want to acknowledge the staff at the Microbiological Lab at both QEH, Best Science and BRL, and Dr. Forge, members of the IPC team, and those who collaborate with us in task force, both in animal and environmental health. We all work together, and I'm sure that as we come together to show these results, that we will see better outcomes for patients, better improvements in service, and we all will be very proud and pleased that we can have a hand in this work. This is my team, not my team. This is the team here that will be present with the Prime Minister at the launch of the Global Youth Group. And I want to thank you all for your time and for your attention. And invite any questions.
Thank you. Thank you, Dr. Blaze, for very, very interesting discussion on AMR at the Queen Elizabeth Hospital. Just one comment before I go to questions. I noticed that there was a 59% sensitivity to ciprofloxacin. Is that correct?
59.
Or 59?
Let's see. You talking about urines?
Yeah.
65.
Oh,
65.
65. Okay, that's still not too bad.
No, it's not bad. But
it still lends yourself to perhaps thinking in terms that if that is coming into the hospital from the community, that for sure it is time to look at antibiotic stewardship program for our general practitioners in the community who are prescribing antibiotics.
right.
So
this
I
is-
kind of think
this-
those results will lend itself to perhaps time to initiate that program for the community.
Right. So the hospital does urine for everybody. So this is a mixture of both inpatient and outpatient urine.
Okay. All right,
so
thank
this
you.
is-
So let us move. Are there any questions, please? Any questions?
Yes, we had a comment/question from Suzanne, who said it was a very interesting presentation, Dr. Blades. She also is making reference to the high rate of MRSA you mentioned that occurs in Barbados, which she presumed is from patients at the QEH. She wanted to know if there was any idea whether or not these isolates were being examined, if they are going to be checked out for the mechanism of resistance. And she also wanted to know if you knew if it was either hospital strains or community acquired strains that you were seeing with these high numbers.
Right. So I would want to say that thanks, Suzanne, for the question and the comments. Dr. Gittins did 2 studies, I think. Dr. Gittins made a comment on it about categorizing the MRSAs. And I think that they were both community acquired coming into the hospital. And perhaps Dr. Gittins may want to make a comment. Is she still there?
Yes, I'm still here. Thanks.
Yeah.
Just want to say yes, that the majority of the MRSA that was seen in the hospital environment was actually from the community. So there wasn't any specific hospital acquired MRSA that we saw. There's only one instance of it, but primarily the MRSA that we were seeing was community acquired, whether it was in the community proper or in the hospital.
Right.
We were able to determine, well, we were only able to look to see if it had the MET-A gene, and then basically to confirm that it was staph. So they all had the MET-A gene and the PBL gene, which is usually associated with community acquired MRSA. And so we were able to determine at that point. Anything else we have not been able to do so far.
Right.
Okay,
So the community drives the hospital numbers. Thank you.
Okay, are there any other questions?
Yes, Julian Harrell here. Really fascinating talk, Dr. Blaze. So I found it interesting, well, a couple of things. One, your fecalis and your facium data is almost that they're universally susceptible to vancomycin. So you seem to have very, very, very low VRE. And then the second question is, do you have, or have you sort of matched the susceptibility data with actual patient outcomes and actually see whether or not the changes in susceptibility over time is influencing patient outcomes?
So the first question, yes, we haven't got any real vancomycin resistant traps. Sorry. We've had one, but I don't think that came up to be so. We had this again, mixtures of cultures. And whenever that happens, you have to go back and verify that you have this thing. So, but it's not common, probably. We haven't got any. And the antibiogram shouldn't have any in it. And then sometimes when we have these strange bugs, yes, Dr. Ford and his team does monitor the patient outcome, and they try to make sure that the best drug, the best antibiotic is used, so that when the patient gets treatment, you have a very positive outcome. I don't know, I can't say for sure, but I'm sure that happens, what the actual rate of survival is. But I know that when the lab gets the report out, that they work on it rather quickly so that the patient can have as good outcome as possible. So yes, we don't have any VREs, and we do try to get results as to the doctor as fast as possible so that they can have the input in patient care.
Thank you.
Thank you. Are there any more questions for Dr. Blades?
No further questions.
If not, I say again, thank you so much for a most interesting presentation, Dr. Blades.
Thank you, Dr. Lewis, and please, I have to step away. My apologies. Thank you very much.
Fully understood. Thank you.
Thank you.
Okay, we're moving on. I think we keep in very good time here. And it is truly my honor and pleasure to introduce our featured speaker for this morning, Professor Julian G. Hurdle. Professor Hurdle is a leading scientist in the antimicrobial resistance field. He's a professor of microbiology in the Institute of Biosciences and Technology in the Department of Translational Medical Sciences at Texas A&M Health Science Center. He received his BS degree from the University of the West Indies Cave Hill campus in Barbados, graduating with first-class honors in biology and chemistry. He obtained his PhD in microbiology from the University of Leeds, United Kingdom, and undertook postdoctoral training and research at University of Tennessee Health Science Center and the St. Jude Children's Research Hospital. His translational research characterizes the genetic mechanisms of antibiotic resistance and their impact on treatment outcomes, while discovering innovative therapeutic concepts to counter antibiotic-resistant bacteria. His research is published in leading peer-reviewed journals and is credited with the recent breakthrough explaining how the evolution of antibiotic resistance in Clostridioides difficile contributed to the pandemic spread of this pathogen and its association with poorer clinical outcomes in patients. It is truly an honor to present our featured speaker for the morning, Professor Julian Hurdle, who will speak on Clostridium difficile, an enigmatic pathogen in hospitals. Let us give Professor Hurdle a warm welcome to the CACM Symposium.
Thank you.
Are we ready to start? Or are we going to wait a couple of more minutes? Good afternoon and welcome back to the afternoon session of our one-day symposium for the 19th annual symposium of the Caribbean Association of Clinical Microbiologists. We are ready to begin with our first speaker for the afternoon. And our next speaker is also our featured speaker for the afternoon. And we are so honored and fortunate to have her speak to us this afternoon. And I would like to introduce to you Dr. Jodian Pinckney. Dr. Pinckney is currently an infectious disease physician at the Massachusetts General Hospital and an early stage investigator devoted to addressing racial and ethnic disparities in maternal mortality related to vaccine preventable diseases. She read for the MBBS degree from the University of the West Indies, Faculty of Medical Sciences, Jamaica, and received her DM in internal medicine in 2011. Dr. Pinckney completed her fellowship in infectious disease from Massachusetts General Hospital and Brigham and Women's Hospital in 2022. Prior to completing her fellowship, she completed her internal medicine residency at the University of South Carolina, where her research focused on disparities in HIV and hepatitis C testing. Her commitment for addressing health disparities occurred during her tenure as the clinical coordinator for the Center of HIV, AIDS, and Educational Services in Jamaica, where her research highlighted disparities in the awareness of HIV prevention measures between women living in high-income versus low-income settings. Dr. Pinckney has published extensively in leading peer-reviewed journals and has received numerous awards and honors for her work. At this time, I would like to present to you our featured speaker for this afternoon, Dr. Jodian Pinckney, addressing the topic, Factors Associated with COVID-19 Vaccine Uptake Among Reproductive Age Women in Jamaica. Dr. Pinckney.
Thank you so much for that wonderful introduction, Dr. Lewis. Are you hearing me okay?
Yes, I can, and I hope I pronounced your name correctly.
Yes, Jodian Pinckney. Everyone just calls me Jody. Let me try and make sure that this screen is showing up properly. And okay. All right. So thank you again for that wonderful introduction. I make sure this is working okay. It seems to be up. Well, that's okay. I have no disclosures currently. Today's talk, I will go through the background of COVID-19 vaccination uptake in Jamaican reproductive age women. I will talk about my research in this area and future directions. So first of all, why Jamaica? As that lovely introduction detailed, I was born and raised there. Those are my parents, and that's my sister. This was last Christmas, enjoying the beautiful weather. It's where I went to medical school. I did my internal medicine residency there. And then, as mentioned, I was working at the Center for HIV/AIDS Research and Education Services, which is located at the University Hospital, Mona campus. And I really thought I was going to pursue a career in research for HIV. But as luck would have it, or fate would happen, I happened to start my infectious diseases fellowship in July of 2020. And we know that was right in the midst of the COVID-19 pandemic. So as an infectious disease fellow, I was really struck by a case I saw, and we will call her Mrs. X for now. She was a 32-year-old Jamaican woman who had moved to the US just 6 months before our encounter. This was her first pregnancy. She was unvaccinated against COVID-19 because she was really afraid of the side effects that it may have on her fetus. She ended up being hospitalized with severe COVID-19 pneumonia. She required intubation. She had to be paralyzed and prone because her respiratory failure was so bad. She had received inhaled nitric oxide, dexamethasone, remdesivir. She eventually ended up having a tracheostomy and a PEG tube. She had an emergency cesarean section at around 34 weeks gestation, and thankfully had a very healthy infant. But unfortunately, Mrs. X continued to deteriorate and eventually died. And so that was one of the cases I saw that really got me very interested in vaccine decision-making in pregnant women. And I was reflecting on what actually happened to pregnant women in Jamaica during COVID-19. And so this is a graph showing the Jamaican maternal mortality rates between 2000 and 2021. And on the y-axis, we have maternal deaths per 100,000 live births. And on the x-axis, we can see the year. And what was quite striking was that the maternal mortality rate in Jamaica jumped from 99, which it had been on average for a number of years, all the way up to 213. And that was actually the highest maternal mortality rate in Jamaica for over 100 years. 44% of the maternal deaths were attributed to COVID-19. And this was despite COVID-19 vaccines being available in Jamaica since March of that year. Just for comparison, because I am practicing in the United States, looking at the US data, we see that the maternal mortality in the US increased from 24 to 33, which is still very high for a high-income country, but still nowhere near the 230 maternal deaths that we saw in Jamaica that year. In fact, speaking to one of my Jamaican obstetrician and gynecology colleagues, she mentioned that she saw more maternal deaths in 1 year than she had in her entire 15-year career. And so what did we know about COVID-19 and pregnant people in 2021? We knew that pregnant people with COVID-19 were 15 to 22 times more likely to die compared with pregnant people that did not have COVID-19. And apart from death, there were also other very poor outcomes, including preeclampsia, preterm birth. They had a higher risk of stillbirth, and their infants had a much higher risk of neonatal ICU admission compared to women that did not have COVID-19. When we look at the vaccines, we know that COVID-19 vaccines significantly attenuated these risks, although efficacy changed with the evolving variants over time. And we knew that the vaccines had no adverse safety outcomes, that they were effective, and that there was no difference in fertility rates between women who were vaccinated versus women who were not. Despite the efficacy of the vaccines, we knew that in high-income countries, reproductive age women had low uptake of the COVID-19 vaccine. For instance, in 2020 in the UK, they surveyed 55,000 Britons and found out that the 18 to 34-year-old women was the subgroup that was most likely to decline COVID-19 vaccination, with many citing concerns about fertility. And then among reproductive age women, pregnant women had the lowest uptake. This was demonstrated by a study in the US in February 2021, where they were looking at reproductive age female Tier 1 healthcare workers, and Tier 1A meaning that these were priority individuals for vaccination. And most hospitals at that time had instituted a vaccination mandate for their healthcare workers to remain employed. So even in that setting where, it's a healthcare setting, they're highly educated, mandates are involved, we see that the pregnant healthcare workers had the lowest uptake, 66%, among all reproductive age Tier 1 healthcare workers, followed by individuals that were trying to conceive, individuals who were breastfeeding, and then the highest uptake was among women who had none of those. When they looked at the US overall, so this is not just confined to healthcare workers, but all pregnant people in the US, you saw that vaccine uptake was very low in January 2022, with just 41% of pregnant women in the US reporting COVID-19 vaccination. And there were stark racial disparities present, with Black women having the lowest uptake at that time of around 25% compared to the other racial groups. So why was vaccine uptake low in reproductive age women? What were the reasons? One of the top reasons we can think of is vaccine hesitancy, which was actually deemed a public health threat, or one of the top 10 public health threats by the World Health Organization in 2019. And it is defined as a delay or refusal of vaccines despite their availability. There are multiple contributing factors. They have different frameworks to describe these factors. One is the 3C models, where they determine that it's vaccine uptake is affected by confidence, convenience, and complacency. And the other framework used is the vaccine hesitancy matrix, which looks at broader scopes, including the contextual atmosphere within a country, in conjunction with individual vaccine-specific and disease-specific factors. In the US, they've actually tried to move away from the term vaccine hesitancy, and the preferred term is vaccine confidence, which is defined as a level of trust that people have in vaccines and those who administer them. And the main reason for this pivot was to shift the weight of the responsibility of the vaccine decision-making process from an individual or a person to the broader societal realm. So making the healthcare providers, the information that we provide, the entire system more responsible for building trust in the vaccines, and therefore promoting vaccine confidence, rather than victim blaming individuals that don't trust the vaccine. There are four factors that are considered under vaccine confidence, and this includes patients' attitudes and beliefs, patient trust, but also vaccine information. As we know, there was a lot of misinformation circulating online regarding vaccination. So the vaccine environment, And it also takes into account healthcare provider confidence. So are the healthcare providers that people encounter very confident in making the recommendation of vaccination? The other factor that came up as affecting vaccine decision-making is medical mistrust. And this is defined as distrust of healthcare providers, the healthcare system, medical treatments, and the government as a steward of public health. And this is really a response to historical and present-day classism and racism in healthcare. It's an indirect measurement for exposure to discrimination in the healthcare system, and it is negatively correlated with vaccine confidence. There are also other factors that are specific to pregnancy that affect decision-making, including gravidity, which we'll talk more about. So whether this is your first pregnancy or you've been pregnant multiple times, the trimester of pregnancy. So, women in their first trimester are probably going to be less enthused about getting vaccinated compared to women in their third trimester. And then there are policy factors such as, are you covered by your insurance? Do you work at an institution that has a vaccine mandate in place? Do you have access to transportation to get to a vaccine site? So those were factors that were present in the US among reproductive age women. And I really wanted to know, were similar patterns present among reproductive age women in Jamaica? So looking at the Ministry of Health data on ComCare, we found that for women aged 20 to 49, around 53% of them were partially vaccinated. And this was still in January 2022. And then only 25% of them were fully vaccinated. But it didn't have a breakdown of whether vaccination rates differed by pregnancy status, or whether they were breastfeeding. It was all aggregated as one. And in terms of the reasons why Jamaicans were declining vaccination, overall, the number one reason was they did not trust it, according to some public polls that were going on at the time. So that led to my research, which aimed to look at the percentage of reproductive age women vaccinated against COVID-19 across different subgroups. And we also wanted to explore the factors associated with COVID-19 vaccination. And so we did a study, which was a cross-sectional web-based survey of a convenient sample of women aged 18 to 44 years old, including patients, providers, and staff members. This took place at the University Hospital in Kingston, Jamaica. The time period, it was done over 3 months, but it was 3 interrupted months because there was a study interruption. So we had the survey going for the month of February. There was a study pause from March to April, and it resumed from May to June, and then concluded on the 1st of July. The exposures that we looked at, or we were interested in, was pregnancy status. So were things different for people that were pregnant versus non-pregnant? We looked at gravidity, where people were COVID-19 vaccination behaviors different for women that were pregnant the first time versus multiple pregnancies. We looked at reproductive status, so looking at differences between pregnant, breastfeeding, trying to conceive, and other reproductive age women. And then because of the study interruption, we actually had a natural experiment to compare vaccine uptake between 2 time periods. And our primary outcome was self-reported COVID-19 vaccination, which we measured as receiving 1 or more doses of either an mRNA or an adenoviral vaccine. We also looked at the medical mistrust factors, which were operationalized as vaccine confidence on this survey, using question items such as COVID-19 vaccines are safe, and then participants would rate, agree, or disagree on a 5-point Likert scale. Similarly, for government mistrust, We measured using items such as the government cannot be trusted to tell the truth about COVID-19 vaccines. And we measured race-based mistrust using questions such as, when it comes to COVID-19, Black people cannot trust healthcare providers. And these questions were taken from tools that were previously validated in the US. We also looked at covariates such as age, education, comorbidities, and insurance status. For statistical analyses, we did bivariate associations between the sociodemographics and the COVID-19 vaccination. And we did a multivariable modified Poisson regression to estimate prevalence ratios for our COVID-19 vaccination outcome, adjusting for age, education, and comorbidities. So that brings us to our results. The first set we looked at was just for February, and we looked at pregnant versus non-pregnant. At that time, we had 192 participants, 72 were pregnant and 120 were non-pregnant. In terms of differences in sociodemographic characteristics at baseline, we saw that the pregnant women were significantly younger than the non-pregnant women, 35 versus 30. We also saw that non-pregnant women were more likely to have college education or higher, 80% versus 56%. And non-pregnant women were also more likely to have a comorbidity compared to pregnant women. In terms of the vaccine types, most women had received either the AstraZeneca or the Pfizer vaccine or a combination of both. With regards to our primary outcome of COVID-19 vaccination, we saw that it was 74.6% for non-pregnant women compared to 34.8% for pregnant women. So significantly lower vaccine uptake among pregnant women in this cohort. And when we ran the multivariable regression, adjusting for age, comorbidities, education, the variables that remained consistently significant for predicting COVID-19 vaccination were pregnancy, education, vaccine confidence, and government mistrust. Those four were significant predictors of COVID-19 vaccine uptake in the cohort. When we looked at other things such as self-reported trusted sources of information, we saw that healthcare providers were the most trusted group, both for non-pregnant and or pregnant women, with 65% trust in that, compared to just 27.8% trust in the government. Looking at gravidity, which we looked at from May to June, we see that multigravida pregnant women or women who have been pregnant 2 or more times were significantly less likely to be vaccinated compared to women who were pregnant for the first time. And this is consistent with US findings that have found similar things, even though the reason is unclear. One of the reasons given is that primigravida women get more attention at their prenatal visits, antenatal visits, pre-educational sessions, where they may have more vaccine counseling done, whereas multigravida women are kind of like, oh, this isn't your first rodeo. They are kind of left on their own for more decision-making processes. That's a postulation, but more needs to be done there. There was really no difference in government mistrust between multigravida and primigravida women. Both were pretty neutral about whether or not the government could be trusted. When we ran the regression model and included the medical mistrust factors, vaccine confidence, government mistrust, and race-based mistrust, what we saw is once we adjusted for vaccine confidence, there was no difference between the multigravida and the primigravida group in COVID-19 vaccine uptake, which indicates to us that vaccine confidence is really mediating that relationship. And so if we could improve vaccine confidence in multigravida women, there would probably be an increase in COVID-19 vaccine uptake in that group. Similar to the first study, healthcare professionals were the most trusted source for both the multigravida and primigravida women. Next, we looked at pregnant versus breastfeeding versus trying to conceive, and we found very similar results to the US, where the reproductive age controls or other group of women had the highest uptake, followed by individuals who were trying to conceive, followed by breastfeeding, and once again, pregnant women having the lowest uptake among all 4 groups. With the time interruption, we looked at trends in COVID-19 vaccination between the 2 time periods. And what we found was encouraging. We saw that while the COVID-19 vaccine uptake in February was just 35% for pregnant women, it went all the way up to 60% for the months May to July 2022. And that was a statistically significant difference. There was no statistical significant difference in the non-pregnant women group. And the reasons, there are postulations for why this increased, but one of the things that started at the antenatal clinic at the University of West Indies were daily educational sessions that were held in the waiting area on vaccination. So we're not sure if that helped to impact it, as well as increasing safety data in the general societal atmosphere. So in conclusion, we saw that self-reported COVID-19 vaccination among reproductive age women varied based on subgroup, with the pregnant subgroup having the lowest uptake. We saw that within the pregnant subgroup, multigravida pregnant women may be particularly vulnerable or a lower uptake compared to primigravida women. We saw that medical mistrust may be contributing to low uptake, particularly vaccine confidence and low vaccine confidence. And we saw that healthcare professionals were consistently the most trusted source. So in terms of next steps, I think we need to amplify the voices of the trusted sources. So providers, both OBGYNs and microbiologists and primary care physicians, pharmacists, everyone needs to be engaging with their communities and showing up, not just in a time of crisis, but actually showing up to the health fairs and showing up to the church fairs to engage with the community and build that trusting relationship. I think various organizations really need to establish a vaccine champion or a vaccine ambassador that is keeping abreast of the safety information for the various vaccines that are coming on board. We need to bring the vaccine to the people. So, people that may be at work or may have childcare responsibilities, making sure that you have other alternative ways for them to get vaccinated apart from coming into a hospital or a doctor's office. Standing orders, meaning like having vaccination orders present at the visit across multiple sectors, has been evidence-based, shown to improve vaccination rates. And then educational videos have also been shown to improve vaccination rates, as well as dedicated trust-building appointments between the patients and the providers. And I think these things are important to think about now for COVID-19, but also on the broader scale, because there are many maternal vaccines on the horizon. I just went to the Infectious Disease Week conference that was held in Boston last week, and they spoke about RSV, which is now approved for pregnant women, FDA approved, but there's also Group B strep. That vaccine is coming on board, malaria, as well as cytomegalovirus. And so I think one of the biggest things that we learned during COVID is that the healthcare professionals need to be prepared with the safety data by including pregnant people in the clinical trials, so that the healthcare professionals can feel very comfortable recommending these vaccines to the pregnant cohort. I believe there needs to be more multi-sector collaboration. So pregnant women or pregnant people are not just relying on information from the obstetrics and gynecology team, but they're relying on information from their nurses, from the pediatricians, their microbiologists, their pharmacists. And so really having a collaborative effort all around to improve the information available that they can provide and support these pregnant women. There needs to be more mixed method studies to understand the role of the social determinants of health in vaccine behavior, such as childcare responsibilities, internet access. And then overall, I do believe the Caribbean needs to have an electronic maternal immunization registry where we can really track the vaccine uptake in the pregnant population over time. And that will help us to detect early trends and plan very effective strategies. But that is the end. I'd like to acknowledge all of the people that helped with this study, including those that were at the Massachusetts General Hospital, the RAND Corporation in California, and our colleagues at the University of the West Indies that were on the ground, specifically Kamali, Carol, Lenroy Bryan, and in Giovanna Whitto. And that is our Twitter page, and these are references. Thank you so much. Do you have any questions?
Thank you again, Dr. Pigney, for gracing us with your presence this afternoon and such an interesting presentation and very, very, very interesting research on pregnant women. Thank you for that presentation. Are there any questions?
There are no questions, but I would like to thank Dr. Pigney myself for that very informative presentation. It was very good. I really enjoyed it.
Thank you. Thank you so much.
Okay, any questions in the chat, Dr. Glendy? No? Okay, thank you again for your presentation today. Indeed, excellent.
Thank you so much for having me.
Okay, we're going to move on from Jamaica to Trinidad. And at this time, I would like to introduce our next speaker, well known to all of us in the microbiological arena here in the Caribbean, Professor Apaka. Professor Apaka is currently a professor of microbiology and head of the Department of Paraclinical Sciences at the University of the West Indies, St. Augustine, Trinidad. He serves as an honorary consultant microbiologist for the North Central Regional Health Authority in Trinidad. He completed his MBBS degree at the University of Nigeria Medical School and completed his DM in microbiology at the UWI Mona, Jamaica in 2004. He pursued a research fellowship at the University of Toronto, Canada. His current research interest includes a molecular characterization of ESBL, gram-negative bacilli in selected areas of the Caribbean, as well as genome sequences of Streptococcus agalacti isolates in Trinidad and Tobago, among others. He continues to present papers at international conferences and has published extensively in peer-reviewed journals. In addition, he is also the author of chapters in a number of textbooks. At this time, I would like to present Professor Apaka, who will speak to us this afternoon on AMR, Where Are We in the Race? Professor Apaka. Professor, we're not hearing you.
Okay. All right, you can see my slide?
Not yet, sir.
Oh boy.
Take your time. We'll get there. Okay, we can see your slides now.
Well, thank you, Dolores. I'm glad you say we'll get there because, you know, we are not like these new age folks who are so technically advanced, you know. If this was to be done with overhead projector, maybe there would be much experience from us in that aspect. But I hope everybody can hear and see my slide right now.
Yes, we can. Thank you.
All right, thank you again. Well, Dolores, we've all come a long way, and thank you for that introduction. Like as you did mention, I am not a stranger when it comes to the CACM. It dates back to 2004, so that's a long time. So probably in the next 1 year, it would have been 20 years. And I must say that I am always thrilled to be part of CACM because, as you do know, charity always begins at home. You know, whether we go east, west, north, or south, will not forget the days of early beginnings. CACM has come a long way, and we are hoping that it will continue to wax much stronger when some of us would have left the scene. So I was invited to discuss briefly antimicrobial resistance. Where are we in the race? So as you can see, it is not a topic that is based on research, but based on almost everything that we have been saying since we started these sessions in the morning. And so I'm approaching it from that angle. Now, I do not have anything to declare, but only to say that I've always been supported by the people that I work for, whether it is at the hospital or at the university. And so whatever credit that should be given should go to my employers, employees. So they are the ones that will take the credit for whatever we have done. But in terms of any other financial gain or whatever, there is none to declare. And even though I will give credit to them, they do not have any say in what I'm going to share this afternoon. Now, my topic is where are we with the race? So you can see that antimicrobial resistance, which is the race, and then of course the discovery, which is the chase. These two are very much in the two-way race. And who is going to get going to get to the finish line first? Is it the humans that are doing everything humanly possible in trying to stop antimicrobial resistance, and at the same time, they are involved in fueling their enemy or helping their enemy? Or is it the bug itself that is our enemy that, as we're going to see, have developed a lot of wings? So they are not only walking now, but they are flying, as a lot of us would have heard since morning when we've been talking about antimicrobial resistance, which is part of the emerging infections, that is a emerging problem that we're having with infections. Now, antibiotic discovery which is the race, we see it didn't start today. It didn't really start in the sense that as far back as 1910, antibiotics were already discovered, right? And it wasn't, as at that time, either in ancient Egypt, where the first, in Nubia, where the first discovered tetracycline, or even in Chinese medicine, where modes and herbs are coming into picture. So you could see that it is not something that started. It predicts a lot of things. And then when you look at this picture that we're seeing here, you see there are quite a lot of antimicrobial agents that has been in the use. And unfortunately, it was in 2013 that the last class of antibiotics were discovered. But you see that at the time when antibiotics were being discovered or being used, the resistance, the enemy has already popped up. They have already popped up. So it is something that the race and the chase has been going on. And what you could see now, that the roadway to antibiotic discovery has so much, very much narrowed. And if care is not taken, we're going back to the pre-antibiotic. In fact, literature and many write-ups have already indicated that we are almost at the pre-antibiotic era, when there was nothing to treat. Not that they were not there, they were there, but they hadn't been discovered. So something really has to be done. I'm very sure the majority of us on this call is very familiar with the picture that we have been displayed. Now we look at our left, we see it's a plate that shows that the AST, the susceptibility test, is what we would want to see in the lab. Very susceptible to the isolate. And then on the right, we see that even the ones that we may think are susceptible or sensitive, they are nowhere. They are nowhere. So that is the kind of situation that we are finding ourselves, that our hands are being tied. And I am sure, like as I said, this is very familiar picture in most of our labs. Now, there was a report that was given by the Global Research on Antimicrobial Resistance Project. That they carried that study in 2019, but it was in 2022 that they reported it in Lancet. And in that study, they had used, it was a literature review in which they looked at several systems, several reports from countries, and they looked at 204 countries and territories, and they were able to report that there were virtually about 23 pathogens that are linked to antimicrobial resistant deaths. And even 88 of those pathogens, when combined with drug combination, that they have resulted in their high rate of mortality. Now, they reported that 1.7, approximately 1.27 million deaths are being encountered or experienced worldwide. And Again, in that their report, there is an associated 4.95 million deaths. Folks, these are not trivial numbers. And you can imagine if we're looking at those 1.27 million deaths worldwide, if we were to bring it down home to Trinidad, would have been all wiped out. Why? Because our population is about 1.3. Thereabouts. So then again, in that report, they looked at HIV, AIDS, and malaria estimated deaths. And we would think that those would have been causing a lot of mortality, especially in the tropical areas. But in that report, they were able to say that only 860,000 of HIV/AIDS infected individuals, or 640,000, were actually dying from those infections. So if you are to do statistical analysis of 860,000 and 1.27 or 4.92 million, I'm sure you would agree with me that is significantly that calculation will be very, very significant. So again, it means that something has to be done. Now, when we even look at the burden of antimicrobial resistant estimate globally, as you can see in this slide, you see that the majority of the burden lies in sub-Saharan Africa. We do also have from the Asian continent. In fact, those two regions are really competing. And then there was a study that was reported by One Health, which was done in 2013. Some presenters have already mentioned or talked about it, that right now it is estimated that 700,000 deaths occur, all right? But in 2020, in 2050, that number is going to go up to 10 million. I remember Dr. Kohal, when he was addressing us, we might think that 2050 is still a long way, centuries to come, but of course not. Maybe some of us, like I do tell my medical students, are listening. I may not be here then, but that will be your problem by 2050. That will be your problem. You had better start thinking of what to do now in order, to stop it. Can you imagine that right now you're having 700,000, and in a couple of years to come, you're going to be facing 10 million. So, folks, and like I do tell my medical students, it's not a joke. Something has to be done, all right? When we look at the impact of antimicrobial resistance, it's economic, it's physical, it's social, and we can't even compare it with what is happening with hurricane. Now, we might think that we are in the Caribbean, we are well accustomed and used to whether it's the hurricane, or whether it is tropical depression in Haiti, or whether it is flooding in the States, or whether it is house being blown off, the roof of house being blown off in Cuba. These are nowhere near the impact that we do get from antibiotic resistance. It's only unfortunate that we don't see this physically. The impact, as you can see, these people being marooned by the flooding or the rains, then that might be why we may think that it's nothing to be concerned about. Even in the economic impact, there was a report by CDC of what is happening in the US right now, or the report that has been, that the economic impact there is so much that antibiotic resistance economically increases the burden, where they are spending over $35,000 billion every year on societal costs, or even in excess of 20 billion when it comes to healthcare costs. Again, that is no joke. Now, what exactly is antibiotic resistance? It's been defined as an ability of this bug to grow when they're exposed to antibiotics, even at high concentration. So they will just shrug off their shoulders that, it's not for me, it's not for my boys. And what would that lead to? Now we do know the several mechanisms where these bugs use in trying to resist the activity of these agents. And either if they would just block the road, block themselves, and so the antibiotic would not enter. Or if they manage to enter, they will push them out without without blinking an eye what is happening. Or if they do enter, they release their enzymes to inactivate them, all right? Or some of them, either by mutation, would just modify the site at which these bugs would act. And then, of course, how are they spread? Locally, we know how it is spread in the hospital, at homes, and the community. It's from person to person, and more so in the healthcare workers' environment. The healthcare workers' hands are the primary agents that are spreading these multi-resistant drugs, resistant organisms. And of course, if we look at it globally, through travel, through agriculture, environmental activities that is going on, and then of course, as we mentioned before, climate change. They are becoming very comfortable in an area where they would have been uncomfortable, but they are now relaxing, multiplying, and causing problems. Well, I took this slide again to re-emphasize to us, especially the students amongst us, to know the common mechanisms by which this antimicrobial resistance occur. All right, and of course, if you look at the reference, you can always consult it to read more. That these agents, they have a way to shortcut, the antibiotics. Some would have three or the four mechanisms operating, or just have one of them, or two of them combined. So there's quite a lot, and we need to start doing something. Otherwise, the 2050 that we're talking about, it will be 2025 or 2030. Now, as reported by that global research antimicrobial resistant group, these bugs, there are the Super 7, and we've been talking about them when Makita mentioned in her presentation, she did talk about these super bugs, the E. coli, Klebsiella, the Staph aureus, Acinetobacter baumannii, or Pseudomonas aeruginosa. And added to that, you have the Streptococcus pneumoniae and the Mycobacterium tuberculosis. The problem with these bugs is that they are armed and they are very dangerous. And unfortunately, we don't see them like the rat running around, but they are there around us. And if care is not taken, when they hit their target, the effect is very, very devastating. They do this with lots and lots of genes which they encode and which has been reported globally, especially whether it is in the Enterobacteriaceae that are very ubiquitous or the gram-positive cocci that are very ubiquitous. Even the carbapenem, that antimicrobial agent that we have, they seem not to be effective in attacking or destroying them. And reports have. Now, in Trinidad, we've not been able to report officially, some of the resistant mechanisms molecularly in terms of when these organisms are using the genes. But I know whether it is NDM, which has been reported in Jamaica, or KPC, that has been reported elsewhere. So this picture gives you a kind of panoramic view of the genes where these are either endemic or whether they are persisting and unabated. Now, the World Health Organization have clearly, either by their studies or by their proclamations or by a transmission, giving us information on the causes of antibiotic resistance. We do have overprescribing of these antibiotics when people just have ordinary cough that is caused by virus. The healthcare providers' instinct is immediately to prescribe antibiotic, even though that antibiotic will be of no use. All right. And in some places, patients can get these antibiotics. They can get it over the counter without prescription, just like what happens here in Trinidad. You can get a ciprofloxacin over the counter without prescription, and that is a time bomb. Patient not finishing their treatment, overuse of antibiotics in livestock. Wow, this is even poor infection control in hospitals and clinic, lack of personal hygiene and poor sanitation, or lack of new antibiotics that is being developed. So in as much as we're having these causes, fueling and spreading antimicrobial resistance, those are also the challenges in trying to compete with the antibiotics in the race. There are no new antibiotics that has been developed, no new class since 2013. And even when we look at the ones that FDA are approving, these are ones that are just modified classes and all that. And when we look at how they are being used in agricultural environment, or even in the consumption, again, the states we've been told that more than 3.5 kilogram millions of these antibiotics are being used on an annual basis. And there was a study that was carried out in 2018 where they tried to look at the drivers of antibiotic resistance. In that study, they reviewed 527 studies. And as you can see, antibiotic use tops the drive. And we're trying to stop the antibiotic resistance. But you see that it is in using it that is posing the problem. And in fact, with that antibiotic use, prior antibiotic exposure was even very significant in causing antibiotic resistance. And then we do have healthcare factors accounting for 53%. Patient clinical history, 47, where more than half of those people who have underlying diseases. And just like the underlying disease may be hypertension, or maybe COPD, or maybe uncontrolled diabetes, fueling, that is also the same kind of silent killers, where if you compare it with antimicrobial resistance. And then, of course, community factors, traveling, animal contact, food and meat and all that. So we're definitely having our hands tied when it comes to engaging the antibiotic resistance in a race. Why? Because the discovery is being hampered. Hampered in the sense that there are so many barriers, which include economic, which include scientific, which include regulatory. In 2017, where it is reported that of 11 new antibiotics that were approved. Now, these are not new class, so please don't confuse that, all right? These are new antibiotics that are coming up apart from the class that had been established in 2007, 2009, 9 are from existing classes of antibiotics. And then something has to be done, and in that, there has to be some incentives and the barriers to overcome. More funding has to be released, and of course, there has to be a push-pull incentives given to the different individuals involved in research and development, or given to the antibiotics companies that are, Now, discovery of new antibiotics in the last 10 years will cost over $1 billion. And these are not the amount you call that the pharmaceutical companies are willing to dole out just like that, because even when they make the sacrifice or gamble and dole out this $1 billion, do you know what? Even before they complete the studies, even before they complete the manufacture of the new drugs, they're already going to experience resistance. So there has been a race and the chase comparison, where you do have the traditional methods, where you do have the bacteriophage, and if you compare what is already in the pipeline, you find that we may not be anywhere in the chase. But somehow we are hoping that with genome mining, with antibiotics, vaccine towards the virulence factors, maybe we would get somewhere. Now, WHO has given a fact sheet concerning antimicrobial resistance. According to the fact that they are putting out there, they are reminding us that antimicrobial resistance is a complex problem. And then because it's a complex problem, approach in tackling it also has to be multifactorial. It has to be approach, so not just through one approach. And antimicrobial resistance affects all of the society, whether it is in their finance, whether it is in their social activities, or even policies, okay? And because antimicrobial resistance is driven by many interconnected factors, one single method cannot be implemented or used intervening to limit so there must be coordinated action required by everyone, by the microbiologists, by the medical doctors, by the lab technicians, by the politicians, by people in the community, everybody, okay? And all countries would have to take a stand. So it's not a war that you will say, oh, it is a Russian war, it is a Palestinian or Israeli war. It's a war that has to affect and be implemented and be supported by everyone. And in that fact also, they have indicated that there is greater innovation and investment that is required, especially in research, and development of antimicrobial agents, or even in producing vaccines. Right now, there's a study that that Wellcome and the Gates Foundation has funded to Sanga Institute, where they are looking at producing vaccines for Group B infections. So we're hoping that maybe in the next 1 or 2 years, the vaccine that is out there now will be supplied to every nation. Maybe that would be the way to go. But even at that, there's still a need to make correct diagnosis by having the proper diagnostic tools. So, how can we control antimicrobial resistance if we're to win the race? Like I did mention, this is a two-way race between the bug and the humans. So there has to be antimicrobial restriction. There has to be political will of individuals, of nations, of establishments and organizations to say, listen, I'm not going to take antibiotics just routinely like that. And then, of course, those who prescribe healthcare providers have to tell themselves that, listen, we have to health educate the patient to maybe eat right, do proper exercise, rest. That would give the body the opportunity to fight back. Of course, there has to be surveillance to identify the carrier of resistant organisms. The hospitals are every corner. There has to be a prevention of nosocomial infection. While doing that, we will not forget the community-acquired infections, because these individuals are coming from the community. Or when they are discharged from the hospital, they will still go back to the community, right? And there has to be adequate hand hygiene. In fact, this should be number one, when we talk about infection control measures, hand hygiene, hand washing, which to some of people feel is a big task. And then for some of us those of us that are in the laboratory arena, laboratory surveillance has to be optimal. In that way, we must try to identify the multi-drug resistance. Now we do the disc diffusion AST method, and then we despise it, thinking that, but that can always give us a clue that when you have multi-drug resistant organisms, that's, and then of course, one of the presenters earlier did talk about this surveillance, stewardship, antibiogram. That all have to be brought to fore, because those are what we can do to try to win the war. Because there are no new antibiotics that is being developed. And as I said before, this is a two-way race. So who is going to win? Are we going to win, or is it the bug that will win? Well, that is a big question, and that is what I am posing to us. Do we want the bug to win by maintaining status quo? Or do we want to win the war or finish the race by trying to tell the antibiotics, trying to tell the bug that we are in charge, we run things, we control. So we cannot just allow you to win this race against us. So folks, this is just a few things I wanted to share with us. Thank you so much.
Thank you. Thank you, Prof, for that very informative discussion on antibiotic resistance and where we are in the race. At this time, are there any questions or comments?
Well, there are no questions or comments in the chat, but my personal comment. Thank you, Professor Akpaka, for that very informative topic on a very topical matter. I would like to say a thank you as well for the work that you and all of our colleagues do in terms of helping humanity to win the race. And hopefully we win.
We definitely will win, come what may, once we apply and do what we can do.
Yes, excuse me? Is someone speaking at this moment?
Now over to you.
Oh, thank you. Thank you, Prof. What I was thinking about is I've been reading a lot and doing some work on another aspect of where we are with the development of new antibiotics. I would like to know what you think. Do you think that artificial intelligence may be the answer to the production of new compounds, as they can screen several compounds within a short period of time to come up with compounds that have activity against certain organisms? What do you think about the use of artificial intelligence and how far they have gotten in terms of being the answer to creating new antibiotics?
Is that for me?
Yes, sir.
Well, it seems that it is the way to go right now. But when we look at it, at using it for as the antimicrobial agent, it wouldn't work. In terms of information, yes. In terms of knowing what would be the appropriate medication, in terms of knowing what would be the appropriate diagnosis, in terms of knowing what would be the effect, in terms of knowing whether it will be effective and all that, yes, artificial intelligence would help. But in terms of what we would take, what we would ingest, well, you know, and AI will not help us in that aspect. But it may help us in telling us that, listen, if you take this, this is how it will work, and this is what will be the end result, you know. But then AI is not going to cut out the hands of people from stopping the intake, you know. So it's going to be something of the future, but not something when it comes to what we would ingest. But if it is acquiring information, knowledge, understanding, yes, yes, yes. And in that aspect, it will really help. Because if you know that you're going to, if you take this medication in a week's time, in 2 weeks' time, this is the effect it's going to produce, or even in 10 years' time, this is what is going to, the effect it will have on your body, you know, then it would have given you a very correct information, you know. But how you now with taking it and swallowing it, it wouldn't help in that aspect. So I would, in summary, I would say, or in answering your question, I would say, yes, it would be the way to go in terms of information that we will get or we'll garner.
So that may be the answer to the 50-year scare.
No, that's not a scare. That 50 years is not a scare, you know, at least projected, just like pandemics are always projected, you know. So we know we are getting there. Yeah, maybe it might be an answer if we do the right thing. Yeah, if we do the right thing, it might really help.
Okay, thank you so much again for your presentation. And we have a few minutes that we're running a bit ahead of time, actually, which is excellent. And at this time, we're going to move on to our friends in Suriname. And I am happy to introduce to you this evening one of the newest clinical microbiologists on the market, actually, Dr. Francis Maui. He's a clinical microbiologist at the Academic Hospital in Paramaribo, Suriname. And we're so happy to have him join us here at the CACM. He received his medical degree from the Faculty of Medical Sciences in Suriname in 2023. He pursued his residency in medical microbiology at the Academic Hospital in Suriname, as well as at Utrecht and St. Antonius Hospitals in the Netherlands, graduating in 2022. He is a current member of the National Immunization and Technical Advisory Group. And today, Dr. Maui will present information to us on the implementation of antibiotic stewardship in Suriname, past, present, and future challenges. Dr. Maui, welcome.
Good afternoon, everyone. Thank you very much for the great introduction. I'm very pleased to be here to share with you some about the implementation of antimicrobial stewardship in Suriname, the past, the present, and the future challenges. Can you see my full screen?
Yes, we can.
Yes, thank you. Thank you very much. I have no disclosure. This is the contents of my presentation for today. I will talk about some general principles and benefits of antimicrobial stewardship. I will share with you the situation in Suriname. I will also share with you some surveillance results of us, and current challenges Suriname will be mentioned, and I will close with some initiatives that has been taken. This is the academic hospital of Suriname, Paramaribo. This is the biggest hospital in Suriname. It is also the hospital with the most specialized departments of Suriname. And it's also the hospital with the only microbiology department in the country where cultures are done with identification and susceptibility testing. This is part of our bacteriology department with some of our lab technicians. Yes. What is antimicrobial stewardship? Antimicrobial stewardship means the using of the right antibiotic at the right time of the right doses for the right duration. And this is easily said than done, but with a few tools, this goal is reasonably approachable. And the primary goal for antimicrobial stewardship is to optimize clinical outcomes while minimizing unintended consequences of antimicrobial use, including toxicity, the selection of pathogens organisms, and the emergence of resistance. With the principle of antimicrobial stewardship, the possibility to obtain quality cultures, and to be able to do that, it is necessary to take cultures, if possible, before start with antimicrobial. But the inability not to collect cultures should not be prevent the start of the initiation of antimicrobial in a critical ill patients. It's necessary to establish a source of control if applicable, and also necessary to written down the indication with all antibiotic orders. And if cultures are known, it is necessary to streamline the narrow spectrum of antibiotics. And the duration of therapy at time of prescribing or immediately following clinical response is necessary to set. One of the tools that I'm personally like to use a lot, that is the 4 moments of decision-making of antimicrobial use. First, does my patient has an infection that requires antibiotics? If I order appropriate cultures before starting with antibiotics, what empiric therapy should I start? And a few days has passed. Can I stop with my antibiotic, or can I change from IV to oral therapy? And what duration of antibiotics does my patient need? And with a good antibiotic stewardship, we are able to improve patient outcomes, reduces the emergence and spread of multi-drug resistant pathogens, and also the good antibiotic stewardship, we are able to reduce the adverse drugs reactions. What is the situation in Suriname? In the academic hospital, we don't have one special electronic patient file for the whole hospital, but the different departments has their own electronic patient device, and that makes some kind of challenges if you need further information about the patient. But as we used to say in Suriname, the country is big, but it's also small, and that means it's a big country, but the doctors in Suriname, we know each other, and that makes it some easily if you need more information that you can take your telephone and call another doctor to ask for information. In the academic hospital, we have a list of antibiotics that we call the list of the reverse antibiotics. And these antibiotics can only be prescribed after written permission of the clinical microbiologist based on indication. And in case the clinical microbiologist is not in the hospital, the infectious disease doctor can also give permission. And for the prescribing of these antibiotics, we have a form that have to be filled by the doctor. And in this form, we have to fill for us the name and the birthday of the patient, the indication of admission, and also the motivation why the doctor want to give one of the antibiotics in these referred list. The meaning of this is not to prevent doctors for giving the patient these antibiotics, but more to look together with us if this the right antibiotic and is this the right indication for the patient. And I can tell that is something that works very well in the academic hospital. But one of the things I will mention later is that in Suriname we have to do with one of the problems that we have in Suriname is not sufficient manpower. And I can tell you at this moment, I am the only clinical microbiologist in Suriname, but from time to time I get support from one microbiologist from the Netherlands. Also in Suriname, we have 6 operational hospitals, but only 2 clinical infectious disease doctors. And that makes it a little bit difficult to adapt the our antimicrobial stewardship that we use in the academic hospital to the other hospitals. But also, as I already mentioned, Suriname is big but small. That it happens a lot that doctors from the other hospital specialists call either the clinical microbiologist or either one of the infectious disease specialists in the country to ask for advice about antibiotic use. And for the list of antibiotics, reverse antibiotics in the academic hospital, the pharmacist is checking the duration of treatments. And I think you will ask yourself if it is in the evening and the microbiologist or the infectious disease specialist is not at the hospital, what will happen with patients who need one of these antibiotics? Some department in the hospital, as the neonatal ICU and the adult ICU, has a small stock of these antibiotics. And if one department need one of these antibiotics, you can say borrow it from the neonatal or the adult ICU, but the next day we have to send the form for us to check if it was the right antibiotics, or is it necessary to change to another antibiotics. In 2017, we had the introduction of the antibiotic booklet. This is WW Antibiotica .SR, and the recommendation in this booklet is based on international guidelines and local susceptibility testing. We have weekly meeting in Suriname with the clinical microbiologists, all infectious disease specialists in Suriname, and we have weekly meeting in the academic hospital with infection prevention expert and also with fellows. But we have to deal with a lot of challenges. Before 2017, all lab results used to put on paper, and as you may know, to give the right antibiotic advice, you need cultural history of patient. And before 2017, it was quite a challenge to have in a short time cultural history of patient. But in 2017, we have the introduction of WUNET. That's a program of the WHO that we could use to as a lab information system for our results. But in 2020, because of WUNET went down, we switched to MyCore. MyCore is a system that they used to use in the Netherlands. It's a system primarily developed to support infection prevention, but especially for Suriname, MyCore has been adapted in such a way that we can use it as a simply laboratory information system. And as I already mentioned, we have to deal with insufficient manpower in Suriname. I already mentioned for a whole country, we have only one active clinical microbiologist at this moment. But and also only 2 active clinical and infectious disease specialists. And one of the challenges that we have these past years also in Suriname, that's a lot of medical expert has left the country. And these medical experts are specialists, general physicians, but also lab technicians and also nurses. One of the other challenges that we have to deal with is insufficient resources. Now I'm going to share some surveillance data with you. But before I start sharing this data, I have some remarks to make. Before 2018, as I already mentioned, data are from paper files. I'm going to share with you data that we analysis of from 2015, 2018, 2019, and 2021. Before 2012, no ESBL test result are known, and the result that the data that I'm going to share with you only limited to blood culture and positive cultures after 2-week interval-free period considered a new episode. I have also some disclaimers to make about the data that I'm going to share with you. Differences between the data that you will see is shown may be due in part to data from different sources or some antibiotic microorganism combination. Numbers are relatively small, which affect the reliability of the results. Differences in procedures and execution and resistant determination in certain periods of time due to temporary shortage of resources. As I already mentioned, one of the challenges that we have to deal with is insufficient resources. As I already mentioned, we have 6 operating hospitals in Suriname. AZP, that stands for the academic hospital, and as I already mentioned, it's the biggest hospital in Suriname. And if you take a look at the blood cultures, you can see the biggest amount come from the academic hospital. And that's not it's not so strange. But if you take a look at the percentage of positive, blood culture, and you can see the academic hospital has the highest percentage, and this may be part due because of too few blood cultures are being taken. And if you look at the LH, that stands for the Slans Hospital, also one of the hospital in Suriname, has the lowest percentage, and that maybe has to do with too much blood cultures that are being taken. Is in a bar chart, you can see the same as what I already mentioned. The clearly higher percentage of the academic hospital AZP and the lower percentage of the LH. You can also see that the biggest amount of positive blood culture come from the academic hospital, as I already mentioned. And the top 10 organisms that are more isolated, you can see in this patch, and the three most highest organisms are E. coli, Staphylococcus aureus, and Klebsiella pneumoniae. And here, from 2015 compared to 2021, the percentage of resistance in E. coli is compared. And if you take a look at for example, at cefotaxime, then you can see a small decline in the percentage. But if you compare this with Klebsiella, you see increasing in percentage of resistance for Klebsiella for cefotaxime. Also, percentage of Pseudomonas. And if you take a look at this chart, you can see it is more or less stable. But what we have the last year in Suriname is that we have total resist to deal with a total resistant Pseudomonas aeruginosa. Same with the Acinetobacter, it is more or less stable, no increasing, no decreasing, but here also we have to deal with total resistance Acinetobacter. If you take a look at the positive blood culture of Staphylococcus aureus, you can see here that in Suriname we don't have a big problem with MRSA, despite the high percentage of positive blood culture for Staphylococcus aureus. You can see it in this chart. In this chart, we compare the percentage of ESBL in E. coli, Enterobacter pyogenes, and Klebsiella. And as you can see here, percentage of in E. coli and Enterobacter are decline, and the percentage of Klebsiella is increasing. Because we, as you can see, in the data that I have just shared with you, it looks like we have a big problem of ESBL Klebsiella, positive Klebsiella in Suriname. And because of that, and because of the fact that we are not able to do further analyzing, we sent the samples to the Netherlands for further analyzing to see if it is one clone not spreading. And as the result came back, the conclusion of the result was there's a large diversity in population, various resistant genes detected, but no resistance to carbapenemase. But here I have to make a disclaimer. These results are from 2015 till 2021. And before 2022, we didn't isolate any carbapenemase resistant Klebsiella in Suriname. But for now, I am sorry to say that, but from 2022 till now, we see frequently, we see a carbapenemase resistant Klebsiella pneumonia, and we are able here to confirm that is the carbapenemase. But what kind of carbapenemase, we don't know that yet, but the samples are collected, and I hope later this year or starting from the next year to send the sample to the Netherlands for further analysis. Current challenges are, as you can see, the ESBL positive Klebsiella pneumonia strains that are spreading through the hospitals. The pen-resistant, total-resistant Acinetobacter baumannii that colonize and infects patients frequently, and the total pen-resistant Pseudomonas aeruginosa that are colonize and infect patients infrequently. But there are several initiatives that have been taken to counter the challenges. As I already mentioned, in 2017, we had the introduction of our national antibiotic booklet based on international guidelines with susceptibility testing in Suriname. Also, the possibility of this booklet is that doctors are able to leave a comment in the booklet, and one time a month, the clinical microbiologist with the infectious disease doctor take a look at these comments, and if necessary or if possible, we give a reply. And sometimes it happens that we invite the doctors for further discussion. In 2018, both a National Antibiotic Committee and a National Infection Prevention Committee were both installed, but because of COVID-19, 2019, both these committees are at this moment inactive. In 2029, we had introduction of antibiotics, of a national antibiotic course. It was the intention that this course would be held one times in 2 years, but because of COVID, the second course took place in May 2022, and now we are planning to have the next antibiotic course in 2024. In 2029, a national antimicrobial resistance action plan was written, action plan 2019-2022, in WHO format. This action plan was already approved by the government, but because of COVID-19, the implementation didn't take place. In 2022, through a funding of PAHO, the clinical microbiologist and the infectious disease doctor, together with infectious disease and specialists from the Radboud University of the Netherlands, had the introduction of a national antimicrobial stewardship program. This program is already finished. I already mentioned that at this moment, we have only one clinical microbiologist in Suriname that gets sometimes support from a microbiologist from the Netherlands. And in June 2023, we start with a training of a doctor to become a new clinical microbiologist. Also in this year, we're starting with an exchange program for lab technician, and as part of that, as I'm speaking now, one of our senior lab technicians is now in the Netherlands for some training, and we have planned to start in 2024 with 2 studies regarding AMR. One of the studies is in collaboration with PAHO that will take place in hospitals, and one of the other study will, in collaboration with an institute from the Netherlands, will take place in the general population, but also we will look to the use of antibiotic in animals. And that was what I had to share with you for today. Thank you for your attention.
Thank you so much, Dr. Mawi, for that presentation and bringing us up to date with what is happening in Suriname with regards to antibiotic resistance, and you're establishing an antibiotic stewardship program for the country of Suriname. It was quite an interesting discussion, and I see, like everywhere else in the Caribbean, that you have a lot of work to do to ensure that we will curb the increase in resistance to antibiotics. Thank you so much. Are there any questions?
There are no questions at this time, but thank you, Dr. Maui, for that presentation. It was very insightful, and I too appreciated the fact that you focused on data from Suriname. It was very informative, so thank you.
Okay, thank you. This ends our actual presentations for this afternoon. And we will now, if there are no further questions, we'll go on to the abstract session. Dr. Glendy, are we ready for that session?
Hi, Dr. Lewis.
Yes. Are we ready for the
Okay, this is Dr. Toms. I'll be chairing that session. We're having a little bit of technical difficulty, so just give us 2 minutes, and we should be ready to go.
In the meantime, I've gotten some phone calls from my Barbadian colleagues with regard to doing the survey. As indicated in the chat, just include your name and email address and indicate that you are from Barbados, because there is no charge required for CMA points for the Medical Council in Barbados. You will see that there's a form that has a charge for Jamaica and Trinidad. Ignore that, please, and indicate your name, that you're from Barbados, and your email, and that's fine.
Okay, while I wait on Dr. Reynolds Campbell, I will introduce our next our abstract segment. It is my pleasure to chair the abstract segment for this year. And this year, we'll be highlighting an abstract for oral presentation that fits right into our theme. Dr. Lydia Miles-Saunders will present a case of Wolff-Hartii-Immonius Chitin iclastica, an emerging cause of gram-negative bacterial sepsis. Dr. Miles-Saunders is one of our DM in medical microbiology candidates here at the University of the West Indies, Mona. She's one of our rising stars. She also has an ASc rather in biology, a BSc in medical technology, an MSc in medical microbiology, and the MBBS. She is a mother of 3 young children aged 2, 5, and 8 years. She enjoys gardening and writing children's stories. Her goal is to impact persons one life at a time for God's glory. Dr. Miles-Saunders, the floor is yours. We are just working on getting her presentation up on screen.
Good afternoon, everyone. Thanks for the opportunity to present this small abstract. It's a case report of an interesting organism that popped up in our department a few months ago, which I've collected data and did some culturing. Dr. Thomas, if I'm allowed to share my screen, can I attempt to see if I can help? Okay.
You're up. Well, if you want to go ahead, you can go ahead.
That's fine. That's fine. Because you're having difficulty.
Okay, the floor is yours, Doc.
All right. Is everybody seeing the screen?
We're able to see.
All right. So as we said, we are going to be presenting on this interesting organism, Wolff-Hartii-Immonius Chitinaclastica. Just hoping that I don't bite my tongue by the time we finish presenting. So the title of my presentation is The Wolf is in Town. And this was chosen because this is the first time we are seeing this organism in Jamaica, well reported, and in the Caribbean. I searched it and I wasn't able to find any literature with it in the reported literature with it in the Caribbean or in Jamaica. This particular case is found in a mentally ill patient here at the UHWI hospital. Okay. Just give me one second. Okay, so just some background information on this particular organism and its relation to humans. So Wolfhartimonas chitinoclastica was first described in 2008 by Toth et al. When she isolated the organism from the Wolfhartia magnifica fly, which we see here in the background. And that is the basis on which the bacteria got its name or its genus association. So the infections so far are closely linked to not just the Wolfhartia fly, but also Lucilia, Chrysimona, and Musca. Those are other types of flies that it has been associated with. And in reading literature, you realize that with global warming and the ability of these flies to survive in areas that they wouldn't necessarily survive on for longer periods of time, including colder areas in Europe, you find that this emerging organism is being found in association with human infections more often than before. So here in Jamaica, though, I could not find any entomology reports of this particular fly. However, I know that we do have quite an incident of meiosis in our patients, especially those in rural settings. Working in rural hospitals, you'd often see patients with peripheral arterial disease, diabetic ulcers, and so on, who would present. And even taenia capitis, you'd see quite a few children in the rural areas with meiosis of the scalp and ears. I've seen it in the ears as well. So the opportunity for patients to develop bacteremia in association with meiosis is a strong one in our setting. Of note, though, the flies most commonly associated with meiosis in Jamaica is a new age screwworm or New World screwworm, Cochleamia hominovorax. And there's no evidence of so far of Wolfhartimonas chitinoclastica with Cochleamia so far. Well, it has not been tested. And there's grounds for research here where that is concerned. Okay, so an analysis done by Arp et al. In 2022 showed that there's often a lot of bacteria associated with meiosis or larval infections in the skin, and swabs done of areas where there was meiosis of soft tissue infections, and also sterile cultures of the organism prove that you have a myriad of organisms that can be associated or found with these organisms. And these the bacteria associated with meiosis would include like E. coli or Proteus mirabilis and Staphylococcus species. And whenever we found the larva, the sterile culture, the larva itself would carry quite a mixture of GNBs as well as GPCs that is commonly found with these organisms. So more specifically, the obligate parasitic Wolfhartia magnifica larva, it develops in live vertebrates. And, it's commonly said that larvae likes dead flesh. That's not necessarily true. The flies are attracted to the smell of the blood and so on. But these organisms actually eat live flesh. It doesn't have to be decaying. It actually will cause necrosis in the flesh and putrefaction based on the bacteria and so on. But it just needs an open wound. The wound doesn't have to be necrotic for the fly to be attracted to it and or for the larvae to eat. So in regards to the organism, the bacterial organism now, Wolfhartimonas chitinoclastica, is a strictly aerobic, non-motile, gram-negative bacilli, and it separates itself out from other aerobic GNBs like your Pseudomonas and Acinetobacter by its strong chitinase activity that is noted within this particular organism. So I have mentioned meiosis before, but I did not tell you but I know most people are familiar with what meiosis is. Just to reiterate, meiosis is a parasitic infection of animals with a diphtheria larvae, and diphtheria larvae is a fancy term for maggot or grub, right? So we know within the life cycle of these flies, they have various larval phases, and whenever this larva along the life cycle lodges in the tissue of a live organism to cause an infection, is called meiosis. Of note, the larvae can also develop in like soft vegetation or any wet or moist, not wet, but moist area like tree barks and so on. It doesn't have to be flesh. So the Wolfhartimonas tritinoclastica larvae you can see here highlighted in the picture causes local or systemic infections originating from wounds infested with the fly larvae. And of note, I'm not speaking about the infection of the larvae, I'm speaking about bacterial infection associated with the larvae. So the infection in humans reported to date comprises of wound infections with the bacteria, cellulitis, there's been a case of osteomyelitis reported, and sepsis. So why this particular case? I'm not sure how many persons would have heard about the organism before, maybe some. But I found it interesting at first when the case came up to us, when it was brought to our attention, not the case itself, but the patient's blood growing this particular organism. And in researching the organism, I realized it was associated with meiosis. And in going through the patient's information and going to the ward to look for the patient, I realized that the patient here in Jamaica had a florid meiosis infection in the right hand or right palm. So the objective was to bring awareness to the clinical community of the presence of this organism in our region and its emergence as a possible cause of wound infection, septicemia, and even death. So the study is just a case study, and it's only one patient so far. Information about the patient was collected from the hospital's patient record and also the hospital information system, where the initial contact was made with the patient in the accident and emergency room. The culture information was also garnered from the laboratory information system, as well as myself as a researcher went ahead and subcultured the organism and did some biochemical workup. So the case report itself is of a 57-year-old patient who is a known schizophrenic or a patient with schizophrenia who was diagnosed with bacteremia with the Wolfhartimonas tritinoclastica due to a maggot-infected right hand. I already highlighted how meiosis is a relatively common occurrence among patients, especially in the rural setting in Jamaica, and those with poor social support or poor hygiene. So this 57-year-old patient was actively psychotic due to non-compliance with his medication. When he presented to the emergency room, the relatives had noticed that he had decreased appetite and was decreasingly active and not as responsive as he used to be. Of note also, they highlighted that he had tried to climb a high fence 2 weeks prior and had received a laceration to his right hand. And a few days before that, they noticed that it had some amount of drainage and the wound itself was worsening in how it appeared. When he came to the emergency room, it was noted that he had lost the 4th and 5th digit of the right hand. Those were completely gone. There was a lot of necrotic tissue in the palmar surface, and when you read the information from the association, the person who first saw the patient, it said that it had copious amounts of maggots to the palmar surface of the hand. The wound was malodorous and it was draining a seropurulent drain as well. However, the patient did not have a fever, did not report any vomiting, headaches, chills, or any other signs of a bacteremia. His vital signs and presentations: blood pressure was 107/62, a pulse of 87 beats per minute, respiration at 22, temperature of 36.1 degrees Celsius, and SpO2 readings of 98% on room air. His initial blood test that was taken included a complete blood count and his renal function. The white blood cell count on presentation was 11.95 times 10 to the 9, and a hemoglobin of 11.2 grams per deciliter, sorry, and an absolute neutrophil count of 10.2. So there was predominant neutrophilia. There was some pre-renal azotemia that I thought was due to dehydration, BUN of 8.2 with a creatinine of 94, and his other electrolytes were normal. Liver function or CRP was not done at the time. All right, so here we are seeing a radiograph of the patient's right hand, and as we can see, there is neither soft tissue nor any bony remnants of his fourth and fifth phalanx. There's just the metacarpal bones that are standing. And if we can see that there's significant soft tissue destruction over on the other digits as well, despite the orientation of the radiograph. I hope we're appreciating that. There was nothing of note really in the chest radiograph, and the patient did not have any significant respiratory findings. There's just some perihilar opacities, heterogeneous, but nothing of mass significance in that radiograph. All right, so for his cultures now, they surprisingly, they cleaned the wound, debrided it, and dressed, but there was no wound culture sent to the lab. However, a blood culture was taken for the patient because of the confusion and the presentation. The blood pressures were a little low, so at first the physicians screened the patient's blood. No other cultures were done, no wound or urine culture, just a blood culture. One set was taken. That was set up to our BacT-Alert automated system. And as expected, the organism, which is strictly aerobic, grew from the aerobic bottle. So one of two bottles came down, the anaerobic bottle did not come down. And that came down within 47 hours of incubation in the system. If you can appreciate the gram stain that I did and took a picture of, these organisms are really tiny. They're small GNBs. This is at oil magnification, and you can see the arrow pointing to one here, and at about 2 o'clock, heading to 2 o'clock, there's another curved rod. Did not indicate that one, but I hope you can appreciate same. So they're very small gram-negative bacilli. Okay, here now on the blood agar plate, and I subcultured it to blood and McConkey agar to note the characteristic or morphological features of this organism. We can see a grayish or more creamish gray, flat irregular edges on the blood agar plate, and it appears moist, not mucoid. There are other papers that would say the organism appears mucoid, but we never got that mucoid appearance. Just it's moist, right? And on the McConkey, we see actually so a non-lactose fermenting organism. All right, so biochemical tubes were done. Our short-line biochemical tubes include our Kliger tube, which had, as we expect for a strictly aerobic organism, which was just alkaline over no change. The urea and citrate tube had no reactions, or they were negative. It's non-motile, and the indole test was negative. And over on our PPA tube, we realized that it is PPA positive. And this reaction is very similar, as I would say, to other non-fermenters, right? So traditionally, if we did not have the MALDI-TOF, which I'm going to speak about, this organism could have easily been misidentified as another non-fermenter. Okay, so just briefly, the MALDI-TOF is being used more widely in the clinical microbiological setting, the principle of this machine, basically, it uses mass spectrophotometry. We have a matrix that is the organism is immobilized on a plate, and matrix is placed over the organism. Then we have a shooting beam that is, or laser that is shooted at the organism or at the immobilized spot on the plate. The matrix prevent complete not just ionization, but disintegration. It prevents complete disintegration of the organism when you shoot the beam, and basically also allows for ionization, which is put up into a tube, or like not more like a photo tube, and in a linear pattern, and then basically it is measured based on the mass per flight, the time of the flight of the various portions of the broken up molecular structure of the organism. The molecular size is now compared graphically to a database, because you're going to have larger molecules reaching at a slower pace than your smaller molecules, and it is separated out into a graphical representation based on the structure of the molecular structure of the organism, and how it basically fragments and is compared in a database, and then an identity is sought or given. I hope that was clear. Right, so the MALDI-TOF is being used for the identity more routinely of our organisms, and it gives a quicker turnaround time compared to our traditional methods, and it is also a reliable method. Okay, so here now I have the original Mueller-Hilton plate that was used for antimicrobial susceptibility testing for the organism. We did not do a method that could give us proper MICs, but what we did do was to do our traditional Kirby-Boyer method with our 0.5 McFarlane standard. So I wasn't able to find any zone diameters in the CLSI or UCAS manuals for this particular method. And even if we did an MIC related antimicrobial susceptibility testing, like tube dilution or so on, what you would have to use in the UCAS is the non-Enterobacteriaceae profile, because there's none specifically there for this particular organism. That's how new the organism is. So for the ones that we did with our disk diffusion testing, we did measure the zone sizes, and the zone sizes were pretty large. So we would basically have to suppose, based on other research that was done with their MICs, you'll realize that the organism itself has a very good susceptibility pattern to or cephalosporins or beta-lactam drugs mostly. And the only paper I saw with the noted gene resistance in this organism was it is intrinsically resistant to fosfomycin. It also had, there was one researcher who found resistant genes to ceftazidime, or it can be resistant to ceftazidime if it carried the BLA VEB1 gene cassette that would give resistance to ceftazidime, ampicillin, and tetracycline together, but that was only found in one particular isolate. We weren't able to do that extensive search to see if there were any resistant genes because no genomic studies were done. But what we did see was that the zone sizes that were given would have been correlated to the MICs in other research papers, and we could safely say that our cephalosporins were pretty sensitive, or anything with a beta-lactam would have been sensitive. All right, so for the treatment in this particular patient, the patient remained afebrile throughout his admission. Initially, he was given roquefin in the emergency room, 1 gram stat, but that was switched to Zinacef 1.5 gram IV TDS for 10 days. He never became febrile after that, and they switched him to oral antibiotics after the 10 days, and he He did 3 days in hospital and 4 days outside of hospital. So he completed a week of oral cefuroxime. So he was discharged without any further deterioration or complications, and his blood cultures were not repeated post his treatment. The patient never had a repeat admission or hospital admission, and so was determined to be completely cleared of his infection, and he was discharged with referral for continuous dressing to his wound. So this is the first case of Wolfhartimonas clitonistica in Jamaica and the Caribbean. It's an emerging pathogen associated with open wound infestations with meiosis. Human infections have been reported in Australia, Europe, and the US, and there's also been association of the organism in infections in animals. It was even found in frozen meat in Brazil, and as a cause of endocarditis in dolphins. So there have been some zoonotic infections as well, and even in a paw infection in a cow. As I highlighted before, because it is non-lactose fermenting, it has oxidase activity, it PPA positive and so on. The organism has been misidentified in the past, and even on the Vitex system, research has shown that those isolates that were positively ID'd on the MALDI-TOF, when it's placed at a Vitex, it is normally misidentified as Acinetobacter iofii or Commomonas testosteroniae. So the MALDI-TOF is by far accepted in research review as the means of testing, and the Vitex is not recommended for testing for this organism. So the geographical distribution of the organism is still being determined, and Jamaica will be added to that distribution as time goes by. So the majority of human cases have been associated with poor hygiene, and notably so because with poor hygiene, you would have the flies surrounding the humans or pitching on their open wounds, or more flies associating with that patient overall. So far, the organisms seem to be susceptible to most beta-lactam drugs, and resistant genes, as I said, have only been reported intrinsically to fosfomycin. The patient had good response in our setting to the use of third-generation cephalosporin, IV and PO. So I already spoke about those genes, and we know that it's an emerging pathogen, an emerging GNV pathogen that has been associated with, there has been 22 human cases so far and 5 fatal outcomes. And that is important to note, that it can actually cause, even though it is susceptible, if it goes undetected, it can cause death in humans. Of note, this organism can cause, single microbial infection, bloodstream infections, and some of the papers that I reviewed, a lot of the patients had polymicrobial infections, but it was noted that this particular organism can cause serious bacteremia and death and sepsis as a single microbe. So our limitations for this, as I said, is a case report. It's not a big study, but there's room for a bigger study in terms of, we can note the meiosis infections that actually have a bacteremia, and then we can go back now and look and see how many of these bacteremia cases are actually linked to Wolfhartimonas clitonistica in the future, so we can actually see what the prevalence of this organism is in this bacteremia. In the future, if I look back at this, I would do a different antimicrobial susceptibility testing method, preferably using the Vitex system or another birth dilution or some method that we can utilize to get proper MICs for the organism. And also, if available, to collaborate to send the organisms for genomic studies to see which resistant genes are being harbored in the Jamaican isolates. I think this is it. I hope I didn't bore anybody to death. And I must say thank you to MedTech Shanice Thomas for helping me to culture and isolate the organism appropriately, and also to Dr. Walker, our consultant, who actually brought the organism to my attention and piqued my interest. Thank you so much.
All right. Thank you so much, Dr. Mel Saunders. That was an excellent, and I found it a very interesting presentation. The floor is now open for questions. And while you think of your question, I would love if you could indicate with a thumbs up or some signal in the chat how many persons have the MALDI-TOF and are using it for diagnostics. And if you can also quickly throw out some new organisms that you have been seeing in your own region or country rather. I know this one is not the only one that we're looking to write up. We've seen Ergotella lentans. That one is probably a little bit more common and noted especially in patients with malignancy, colonic pathology. But if you have anything else that you can share about, that would be very good to hear about. So I can see at least one other person, but that's Dr. Pinkney all the way there in Harvard, in Boston. Is there anybody else with a MALDI? If you can, just let us know. Well, it's been very interesting to see, some of these new organisms, and it does keep us on our toes. Dr. Pinkney also seems to have a question. You can go ahead, Dr. Pinkney.
No, I find it quite fascinating because the first case report that I ever wrote on abiotrophia defectiva with the microbiology department with Dr. Roy Green, it wasn't via MALDI-TOF, but that was what we discussed in the case report, was that with MALDI-TOF, we'll be getting a lot more identification of these rare organisms, because that was the first time abiotrophia was detected in Jamaica, and it was an infective endocarditis case. But with MALDI, the prediction was with MALDI-TOF would be seeing more and more, which we have been seeing more and more of these difficult to culture or fastidious organisms that need certain ingredients or nutrients to thrive. I would say the MALDI-TOF, obviously at MGH's baseline, we do MALDI-TOF on all the cultures. What we also do a lot of is sending for 16 and 18S RNA sequencing in our cases of culture negative endocarditis particularly. And the organisms that sometimes show up even on Carius, which is another like cell-free DNA technology. Carius is even sometimes Haemophilus influenza, which didn't grow. So it's not necessarily a rare organism, but things show up. Yeah, so things show up on these I think I would say probably one of the rarest organisms that I saw that showed up on a MALDI was Schwannella, of a person that had gone kayaking and had just gotten like a fresh tattoo that day. So
okay, we've seen Schwannella as well, so that's interesting. Yeah. Okay, thank you for that. Thank you, and thank you for mentioning sequencing. We have a sequencer here, so Dr. Mel Saunders. Maybe you could look into sequencing your isolate.
Yes, definitely. Yeah, I'm thinking of going. I hope I find it to do the I'm thinking I'm going to do the MIC on it using the Vitek and see if I can probably try and get this published.
Yes. And it's interesting to also see how, sometimes you see these cases of myiasis, we don't often think of the risk of infections that the larvae themselves can cause. So this certainly wakes us up a bit, and we need to be on the lookout when we're managing those cases. We see them from time to time. Don't know if there are any other questions. You have some comments. I don't know if Dr. Williams can help me with these comments. Dr. Walker, I want to commend you as well. Let's see, you have a comment. Dr. Walker says, good presentation, Dr. Mel Saunders. I appreciate highlighting the importance of correlating the clinical picture with organisms that are identified using the MALDI, underscoring that this should be done in cases where rare or unusual organisms are identified. And he agrees with you that the MIC testing would be helpful. He congratulates you again. So too does Suzanne Workman. Very interesting case report. Well done, she says. And Dr. Pinkney encourages you to get it published. Dr. Brown, the floor is yours. If you have a question, you can go ahead. I'm not sure if Dr. Brown can open her mic. Let me see if I can. Hold on. Trying to find you, Dr. Brown.
Yes.
Okay, great. Go ahead.
Thank you so much. So great, Rick, Dr. Saunders. And my question is really just out of interest. I was just wondering if other case studies noted any like co-bacterial infections. And I'm a virology person, I'm a virologist, right? So forgive me if my questions are odd. So I was just wondering if any of those other cases that you may have looked at in the literature had any co-infections, and if that may have led to some of the worst outcomes that some of the patients would have experienced, or even if their wounds were more severe or anything like that. Thank you.
Thank you for that question, Dr. Mel Saunders. I think that's a great question, Dr. Brown.
Right, very good question. Okay, what I did note was that surprisingly, the people who, I need to look more closely at it, but a lot of the people who had fatal outcomes, they were on a combination of zosine and vancomycin. And that's something that while I was reading, I said I need to look more closely at this, because it made me wonder if the combination of what is chosen is important, or how far along the infection was. So one fatal outcome was a homeless person. If I can share my screen, Dr. Toms, I'll go through a chart that the study had 5 fatal outcomes.
Yeah.
So there are only 22 human cases so far, and the 22 were tabulated by a researcher who just went through, tabulated all the case studies and looked at the outcomes. So just allow me to share, and I'll just show the table and show you who actually, all right.
You can go ahead.
Let's see. Where is this one? Are you? All right, I'm not sure which portion of my screen you're seeing.
We're seeing the bit about occlusive peripheral arteriopathy.
All right, let me see if I can adjust so you can actually see. All right, so this is a table of patients who survived or who died, right? So here we have a patient who, let me just make sure this is the first one. So the first patient is in 2009, right? Was reported from France, was a 60-year-old homeless patient who's an alcoholic, right? And the antibiotic use was ceftriaxone. He survived. So this homeless patient survived, but let's go down to the fatal outcome patient. So here we have a fatal outcome in 2011. This is a patient, homeless as well, who is a 70-year-old patient from Argentina, right? Of note, most of these patients had meiosis, right? There was only one patient, I think, who had just very bad hygiene, and they thought that the ulcers on the body predisposed the patient. And they thought flies would have pitched on the patient, but not necessarily or left the larvae. This patient died. There's no indication in the study that the patient was had any other worse underlying illnesses than the other. I think it's where along the infection timeline that it was discovered for the patient. So for this patient, they used the quinolone. They also used ceftazidime and amikacin, which our patient actually got a third generation cephalosporin as well. He also got an aminoglycoside. All right. So we here now we have this patient survived, so we may not be interested in that one. It was in 2015. It was a patient who was having recurrent falls. He had chronic kidney disease, ischemic heart disease, osteoarthritis, and he was found unconscious. Actually had a bacteremia, but he survived. So this patient had a lot of underlying illnesses. This patient was morbidly obese, had progressive gangrene to the lower limb as well, and had the meiosis. He was given cefpodoxime. Again, he survived. This patient got augmentin. And if we realize a lot of these patients have bad social circumstances, some of them homeless, and a lot of alcoholism also related to their infections. So this third patient now, who also had a fatal outcome, this combination of tazobactam, piperacillin, and vancomycin, I realized was there for the use of vanco and metronidazole for this fatal patient. And you realize that the underlying illnesses were not necessarily vastly different, as in to say if, you know, one had worse comorbidities than the other. And then this other fatal patient, well, this one had metastatic adenocarcinoma and immunosuppression. So despite treatment, this patient died, but of note, this patient was also on single drug therapy. And I actually need to look what generation of drug this is. I'm not sure at the moment. Right, but of the 22 patients that they have found human infection so far, those who got double treatment with cephalosporins seem to survive very well, or even with single cephalosporin. It all depends on which one is chosen. Again, we have here a combination of vancomycin and tazobactam that actually, vancomycin and zosine, sorry, that actually had a fatal outcome as well. So I want to go and look at those more carefully to see if the drug of choice played a role. However, this person who did the antimicrobial susceptibility noted that there are not many resistant genes that have been noted in the organism so far. This is coming from animals to humans. It's not, you know, it's not widely exposed to antibiotics thus far. Therefore, when it is exposed, it does respond well, except to fosfomycin, which it has intrinsic resistance. So the drug of choice may not be it, may be other factors, but we're not sure at the moment.
Okay, thank you for that, Lydia. So it will be interesting to hear what you find and really to see if there is co-infection, especially since some of them are on multiple antibiotics, which really don't seem to make the best sense in terms of combination, ampicillin with a third-gen cephalosporin and a quinolone and an aminoglycoside. So perhaps there were several things at play there, but we're happy to know that you're going to explore that some more. Prof. Akpaka, you wanted to either make a comment or ask a question? You can go ahead.
Thank you. What I wanted to do was to lend my support for the call for Lydia to have the paper published or reported, because as she was presenting it, and then I remember in 2008 that we had the first case of KPC producing Pseudomonas aeruginosa. Now, we were able to publish that case report in JCM, Journal of Clinical Microbiology. And in fact, that was the first time I was able to go to ICAC. In fact, that year, 2008, the ICAC combined with IDSA to have a combined annual conference. That was, I think it was in Chicago, you know, and that abstract to report this case was accepted, not just for poster, but for oral presentation. And I was able to make that presentation, and later on, as soon as we came back, we wrote it up. But what was interesting, like now that in Jamaica you have the sequencer, that should enhance the research capability, because we have moved away from all these MIC values and the prevalence values and all that, you know. So Lydia, you should do everything to get the proper analysis of these organisms and submit it for an abstract. Another, well, as a case report for one of these leading journals, and you'll be so surprised they will accept it. That's just what I wanted to say.
Thank you so much for that. Lydia, I believe that is your intention, correct? You do plan to publish. I happen to have insider information. Lydia, are you there?
Well, I'm not sure if it, well, I wanted to, but I'm not sure if it will be accepted because I did not do the MIC. So I only did the disc diffusion. So I'm not sure if I find the organism, hopefully nobody threw my organism away. If I find it, I will definitely do the MICs, and probably to give me a better chance of it being published. I don't know how acceptable the Kirby-Boyer method is going to be, seeing that most people are using their fancy MIC methods now.
And maybe even try to see if you can get it sequenced. But we will look to see how far you get. Thank you, and please join me in thanking Dr. Miles Saunders again for an excellent presentation. Mine is also the pleasure to present this year's CACM Awards and to take us to the end of today's session. We have awards for the best student and the best overall abstract presentation, long service awards, and for the first time this year, the Medical Microbiologist of the Year award. I will now read the award citations and the winners. I'll be assisted by Dr. Mark Williams. The first award, the Shivnarine Kissoon Youth Microbiologist Award for the Best Abstract Presentation at a CACM conference by a student member of the CACM. The late Shivnarine Kissoon joined the association in 2010. He was a dynamic addition to the team who left an example of dedication, commitment, and excellence for all to see. It is hard to speak of our late colleague without mentioning his unbridled commitment to his Christian faith. He somehow managed to combine both microbiology and his Christianity without discrimination or compromise. He had an infectious sense of humor, which made him very good company. He's greatly missed and can never be replaced. The student to be awarded must be a CACM student member studying microbiology or related field who has not yet earned a doctoral degree. The award is a waiving of any applicable membership fees for 1 year and a certificate. This year, the Shivnarine Kissoon Youth Award is being awarded to none other than Dr. Lydia Miles Saunders for her excellent presentation and case report on Wolf Hartiaimonius Chitina iclastica. And I hope I didn't make a mess of that. So congratulations, Dr. Miles Saunders. We are now moving to the Surjapat Perisuna Award for the best overall abstract presentation by a CACM member at a CACM conference. So this is for the overall abstract set of presentations, not just for students. Mr. Sunath was the hospital district manager for the Caribbean region for Wyeth Pharmaceuticals, and he came up with the idea of and encouraged the development of an association for microbiologists in the region. He broached this idea with Dr. Nicholson and other microbiologists within the region, to which persons were receptive. With the support of Wyeth, the association was formed in July 2004, with meetings being rotated to a different Caribbean country annually.
Mr. Senath's hospitality was legendary, and his attention to detail whenever these meetings were arranged was outstanding. Over time, the association grew and currently has members from, including but not limited to, Jamaica, Barbados, Trinidad and Tobago, Curacao, Suriname, the Bahamas, the Cayman Islands, Guyana, Bermuda, Aruba, and Grenada. The Kerry Senath Award, named in recognition of his seminal contribution to the development of this association, is also being awarded to Dr. Lydia Miles Saunders for Best Overall Abstract Presentation by a CACM member at a CACM conference. The award is a waiving of any applicable membership fees for the year and a certificate. And for the first time now, we are presenting our long service awards. We have or plan to issue long service awards for persons who were members and supporters, as evidenced by service on committees and participation in CACM events for 15 years, 20 years, 25 years, and 50 years. If persons perhaps start very young within the association, or relatively young. As you heard, the association was only started in 2004, so we've only gone 90. So we're installing our 15-year long service award category this year. And these are our awardees. We have Professor Patrick Akpako, blazing the trail over there in Trinidad. We have Dr. Dolores Lewis, blazing the trail and has blazed the trail in Barbados. Dr. Madura Manjanath, also doing great work in Trinidad. And Professor Alison Nicholson, who has done great work, and dare I say Professor Alison Nicholson OD, doing great work in Jamaica. And Dr. Karen Wargreen, you heard Dr. Pinkney mention, Dr. Wargreen also had her work on an isolate that was pretty novel, also doing great work in Jamaica. All of these persons were founding members, and we offer heartiest congratulations to them and thank them for their dedicated service. They've certainly inspired me and are part of the reason why I'm a part of the association, which has blessed me tremendously. It's a great way for us to network and for us to learn about what's happening within the region in our field. And the last award for this year, again, this one is being presented for the first time as well, the CACM Microbiologist of the Year Award. And I invite Dr. Mark Williams to assist me with this award by reading the citation. Dr. Williams?
Good afternoon, everyone. So, the CACM Microbiologist of the Year Award. Despite needing no introduction, our awardee for Microbiologist of the Year is an esteemed colleague who has blazed the trail in medical microbiology. She's a professor of medical microbiology, serving as head of the department at the University Hospital of the West Indies from 2015 to 2021. During her time at the university, she has served as not only a lecturer, but a pioneer in infection prevention and control and antimicrobial stewardship. Serving as the chair of the Infection Prevention Control Committee, she has delivered numerous talks on the topic, as well as coordinating several workshops and seminars island-wide. She spearheaded the first infection prevention and control postgraduate diploma program at the university in 2021, serving as its current director. A true academic and an author in her own right, our awardee has published over 80 academic papers with over 36 peer-reviewed articles, 1 book chapter, 45 abstracts, and 4 technical reports. Throughout her tenure, she has also supervised numerous postgraduate students, lending them her expertise and passing on the flame of passion that she has for microbiology. She has served as both an internal examiner for the DM in Microbiology program, as well as a coordinator for the same program. She holds membership in a myriad of organizations. She co-founded the Caribbean Association of Clinical Microbiologists in 2004. She's a member of the governance team of the UE COVID-19 Task Force. She serves alongside international organizations such as WHO and PAHO as the UE representative for the National Antimicrobial Technical Working Group. She's a member of the Scientific Research Council, and to top off her accolades, she recently received the Order of Distinction for rendering outstanding and important services to her home country, Jamaica. It is with great pleasure that I announce the CACM's first Microbiologist of the Year, none other than, and I see her smiling, Professor Alison Nicholson, OD.
All right, we'd like to congratulate all of our awardees. And I'm hoping that I can get Dr. Nicholson to open her camera. I don't know if you can pin her. Dr. Nick, one of our residents, is presenting the award to her. Let me remove my spotlight and let me see if I can put one on her. Right, so there she is. Congratulations, Prof. Nick. We are very, very proud of you. We are very, very happy for you. And we know that there will probably be other amazing persons who can receive this award. You can only receive this award once in every 10 years. But we believe that we have a wealth of persons and there should be some awards coming in the future. So Dr. Nick, I don't know if you want to say a few words. I know I'm laying hands on you suddenly. Let me see if I can assist with opening your mic, Doc. We're not. Hold on a minute.
Yeah, my bad. Sorry.
Okay.
So let me start again. Thank you very much, Dr. Toms. And I'd like to thank the CACM for this award. As you know, the CACM is very dear to my heart. When we started, we had a lot of question signs, a lot of doubts. Certainly I did. And to see that it has lasted all these years and it seems to be growing from strength to strength, it makes me very proud and very comfortable that that microbiology is in good hands and that the younger ones coming up will continue to do a good job and continue to blaze the trail. It is one of those disciplines that I think is coming out from the cold, getting spotlight. Some of the spotlight is not really because of us, it's just because of events, but fortunately, many of us were there to take on some of these events and to capitalize on making them work for us. So I don't think microbiology is a stepsister anymore. Just take a look at our residence room, right, Dr. Toms?
Absolutely.
Look at our residence room and we see growing interest. And I think as persons come to understand more about what microbiology involves and the diversity therein, that we'll see more and more persons, taking on the challenge of digging deep and learning more about it. So thank you very much. I appreciate this award. It will be one of my special awards, especially since I'm, bowing out, exiting stage left. So thank you very much. And I do wish you and the organization all the best. And of course, I'm not gone yet, so I will be sitting in on your conferences and so on as long as the interest remains. So I do wish you all the best. You and the team wish you all the best. And thank you very much.
Thank you,
Thank
Prof. On behalf of the association, I want to underscore the thank you and how much we're proud of you. And I'm saying you're welcome on behalf of the team. All right, so we're actually wrapping up now. Just allow me to fix this spotlight. Okay. All right, so on behalf of our president, Dr. Reynolds Campbell, allow me to say thank you to everyone who participated in any way today. Special thanks to the conference organizing team in Barbados. Can you show them some appreciation in the chat? So ably assisted and led by our president, Dr. Reynolds Campbell, who also did an amazing job. We are really grateful. Special thanks to our treasurer, Dr. Roy Green. When we needed money, we ran to Dr. Roy Green and she really helped us. She assisted us with getting CMEs. Dr. Roy Green, we really do appreciate you. Dr. Mark Williams, one of our newest MOs. He's really been helpful. Pretty much almost anything I've asked him to do to assist with this conference, he has helped. And we are truly, truly grateful. Our presenters did an amazing job. We really want to thank you all. You expertly delivered great content that is very useful and applicable to our field and to the times in which we live. And certainly we would not have had such a great conference without your audience. And so we thank God for an excellent session and look forward to the next symposium in 2024. If you are not yet a member and you see all of these awards that only go to our members, we extend an invitation to join the CACM to you. There is a membership category for everyone, even if you are not a microbiologist. Send us an email at cacmjamaica@gmail.com if you are interested. Please be reminded to complete the survey, and if you need it, to request CMEs there using the Google link provided, and it has been posted throughout the conference within the chat. So thanks again, everyone, and good afternoon.